So I've been muted the whole time. Good thing I chucked it now. You can't hear because the sub audio is muted. Good thing yeah, so we're gonna test audio real quick. Sweet. Alright, so let's just start. We're on cell communication right now. Stop refocusing. Stop it. It's okay. It's okay. It's okay. All right. I like him in the corner. Then he just gets bigger or smaller. But I don't know what to do with him. Maybe I'll maybe put him in this corner instead. Look at that. He's here. And then he moves. Isn't that cool? Alright, well, let's lock back in. Alright. So, this is true. Multicellular organisms must communicate and cooperate. Duh, of course, right? Sending and receiving signals, converting those signals into response. There is a little, as our AP Bio teacher kind of drilled this in our head like crazy. Autocrine. Alright. So cell target itself. I don't know why you would need to do that, but I'm sure an example comes in. Juxtacrine, cell to cell, membranes that are touching have membrane proteins. Okay. Then short distance communication. So then this would be like let's see, paracrine. Cells secrete signaling molecules in by cells. Synaptic stimulation of nerve cells by neurotransmitters. Goes through receiver, then goes away. Ooh, that's a good analogy. I don't actually know why Snapchat does that. There's probably a reason. I just have no idea. So note that these two are like the molecules communicating with each other directly, connected to each other. This one, of course, it's connected because it is itself. Then there's this one, cell to cell. The cells are actually connected. And then this one's short distance, where they're not connected, but they're very close to each other, hence short distance. Now, long distance, usually you'll get these through like hormones. The endocrine system, so organs create hormones that travel throughout the body. Circulatory system, how your blood gets around. I like my teacher using modern examples because then they resonate with the student body, If you don't know what this video is, he was, is our endocrine system. It's a jump scare. It's what it is. Epinephrine. So there's epinephrine and norepinephrine. Yeah. Okay, so this is the important thing to know. Reception, transduction, response. That's cell communication. It's very important. Reception, it's where your receptor it's the signaling molecule bonds to the receptor molecule, which is called a ligand, and then therefore it starts a transduction where it converts. It sends those through relay signals. You might see those through GTP, GDP, and then eventually all those signals are sent to a response. Transduction, you'll learn about it in AP Psych 2. It's basically, it converts, basically, like, for example, your vision. So it converts those, the activations from your optic nerve and converts it into electricity, which is to be interpreted by your brain. Okay. So ligand, yeah, it's like I said, it's the chemical molecule, or I got it backwards, sorry. Ligand is the signaling molecule, which then bonds to the receptor, which is in the membrane. And then secondary messengers, they're internal proteins that relay messages. Then we'll see more about this later. Cyclic AMP, CMP, we'll hear more about that too. And then, so this is what I was talking about, like GTP. You have GTP, goes to GDP, and then uses ATP to help activate that second messenger, which then does this protein and then makes a cellular response, if we're being basic. Okay, let's see. These pants are too short for me, and then so like the lower part of my leg is getting bothered, so I might leave for a second just to get longer pants, but whatever. Signal perception, intracellular transduction, signal perception, reception. Then you have your cellular response. That looks like popcorn if you think about it. Alright, so reception, as the ligand binds to the receptor protein, changes its shape, therefore changing its function, as we've learned. Then you have transduction, where reception, it sets off a relay of communication throughout the cell, and then this is where the second messengers come in. I'm touching my monitor, sorry, but then see, blah, blah, blah. Then you have the cellular response, which is the cellular response. Alright, so then here's a little graph, if this helps. So ligand's not bonded, right? But then once it is bonded, then this receptor protein turns on, and then therefore, blah, 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 it goes down, and then there's a response. So same signal can trigger different responses depending on the receiving cell. That makes sense. Different cells have different shapes, therefore different functions. Response 2, 2, and 3, crosstalk. So process, like inhibiting, that makes sense. And then, yeah. Each cell has a different set of secondary, yeah, makes sense. Adrenaline. So you don't have to know this, I guess, for the AP bio exam, but it's a good example. And then, as you know, adrenaline is it's part of your what is it called? Your parasympathetic nervous system. You'll learn about that in AP Psych. Promotion of glycogen breakdown, increased heart blood pressure, increased breathing, dilation of pupils, your fight or flight system. Let's see. This is like a technical one. I don't know if you actually need to know this, because in my notes you probably don't. Yeah, you don't need to know. Okay, so interpreting diagrams, just need to know this, right? So, because you'll see this on the AP exam, basically arrows show molecule that's being activated or moved. In the bar-headed lines, it's inhibited. So, for example, pKa will inhibit that. These SUFU will inhibit that. Yeah. Shows molecules that I wrote that is being inhibited. Shows molecule that is being inhibited because I wrote molecules because I'm an idiot. Disorders. Receptor. Okay, so basically if any part of the reception transduction goes wrong, then obviously you have a disorder because your pathway isn't working correctly. Ooh, that's the if you want to know what this is, these are nerve cells, right? And then and then this is the axon. It's what the signal is traveling through, and a damage to the protective wrapping. It's called the myelin sheath. There are some there's diseases associated with that. One of them is multiple sclerosis. You'll learn about that in AP Psych. See, where immune system attacks myelin sheath, protecting nerve fibers, and then because the myelin sheath helps to since it surrounds the axon, it makes it so that signals travel faster and I guess more efficiently, if I remember correctly. So without it, you know, it's a little slower, and I guess by the looks of this, prone to loss. Autoimmune disease, neurotoxins, drugs. These not pathogens. I forgot the word. Not carcinogens either. Some of them are carcinogens, but not in this case. All right, kinases. So kinases, transfer of phosphate groups from ATP to other molecules, and then phosphatases. It removes phosphate groups from proteins. Blah, blah, blah. Okay, let's see. Phosphorylation activates proteins, so deactivate proteins. Kinase. Oh, it's kinase. I even wrote in the pronunciation. Sorry. I actually have no idea what this is. I don't know if I've seen it since then. Interesting. I don't know what part did that. Phosphorylation cascade. Okay, so this is part of the transduction. So series of kinases. I see. This is the kinase. Sorry. Series of kinases, add phosphate to the next one. So it's like it's literally a cascade. I really don't. It's a bucket brigade. Hold on. That's not a real thing. By the way, when I say that's not a real thing, that's usually just me joking that I don't know what it is. activation. Okay. You could think of that like a redstone repeater. Let's see. Water soluble signal molecules bind to specific sites. Okay, so allosteric basically means it doesn't bind at the active site of an enzyme. Let's see here. So you have the ion channel, so it's how it gets through the membrane. G-coupled protein, that's what we were looking at before. I have no idea what this is. I guess we're just supposed to know what it is though, and I wrote it down. Right, we'll talk about here. So G-protein receptor binds with the ligand. G-protein becomes active. Let's see, it's GDP to GTP, and then it's moved and goes to activate other proteins. This is usually an animation, but basically you can imagine this binds here, and then this goes, activates, moves on, blah, blah, blah. Ligand gated. When ligand binds to the receptor, receptor acts as a gate opening, allowing ions through. It's in the name. Receptor tyrosine kinase. As ligand binds with the receptor, tyrosines come together and forms dimers. Internal phosphorylation activates responses. We're going to look this one up because I have no idea what this is. You.
Here we have receptor protein, and it's like the DarkKnight and then state within is to as framer for let's accept that chain to that path is intage level, but have to stick to it.
Okay, so you saw how that works. That's basically all we need to know. Alright, intracellular responses. So intra, meaning within, so like internationals between countries, intranationals within one country. Some nonpolar ligands can pass through the membrane. So usually you'll see it as like hormone testosterone, and then so rather than bonding with a receptor protein within the membrane, it'll bond with a receptor protein inside of the cell. Intracellular responses. Smaller hydrophobic chemical messengers pass through the membrane easily. Ligand bonds with protein within the cell, activates the use of protein. Yeah, that makes sense. Let's see. Additional notes for that. I did not. Okay. I think this was awesome animation. Let's see if we. There's a chance we might be able to find, yeah, here. With Clidio is probably a video, so let's see if we can find that video. Or I guess we're supposed to just label that. So this one, you can tell by the shape. It goes here, this goes here, this goes here. Remember, shape. A structure defines function. Okay, positive feedback, negative feedback. So what you need to know, basically, positive feedback is where the product increases to further production of that product. So I guess like that, yeah. Clotting. So resting and received signal activates more palates, clotted blood activates more platelets. And the negative feedback is where the product inhibits the other one. So, like that, yeah. This one makes sense. So if your body temperature decreases, the heat process is activating, but then as it increases, then it stops because you're at a homeostasis. And then same thing with sweating. Oh, that guy's sweating in his suit. Yeah. Alright, now blood sugar. So blood sugar decreases, then your signal to make more. Blood sugar increases, then your signal to what does insulin do? To store extra sugar as glycogen. Okay. All right, so that's done with. We're done with cell signaling. The biggest thing, you know, I try to go over it briefly. We've already been doing this for about 20 minutes. All right. It'd be funny if, for whatever reason, we also didn't have mitosis in here. The cell cycle. So, there's, okay, so let's see, how long are these notes? Not too long, it's about 8 pages. Alright, so I'm just going to talk them over and I'll bring up, graphs as necessary. So, we already talked about the surface area to volume ratio, that was yesterday. So as the cell grows in size, you know, the surface area to volume decreases, and then so therefore you want to keep the ratio as large as possible, that being more surface area to volume. And then so there's ways to increase that, and that's to divide the cell, to change your shape, or to increase folds along the membrane. So for example, let's take a look at the inner mitochondrial membrane. You'll see, look at all these folds. These folds increase surface area. That's very important. All right. Chromosomes, they're, as you know, they're tightly packed DNA around a protein structure, and parts of the chromosomes here. I don't think you need to know all of these, but you have. So the thing that the chromosome actually wraps around, it's called a histone. So that's what the DNA wraps itself around once it leaves, because originally it's chromatin. And so it's a thin thread of DNA that condenses before mitosis, but then the chromatin comes from the nucleus. It's, you know, it's spaghetti, basically. No, it isn't. Hold on. Oh, so I guess within here, there's already the histones as well. Okay, so now we get to the interesting parts. So we have chromatin, which is the thin strand of DNA, but then you have a chromatid with the D. And then, let's see if I can find where that shows it. Let's see. It's basically an arm of the chromosome. So you see sister chromatids. These two, right? These two are sisters to each other. And then, we don't need to talk about it yet, but homologous chromosomes. I forgot the regular definition, but basically they're chromosomes that are like paired together somehow. Oh, I see. Because see, before replication, these were the two strands, but then once they're duplicated, they're still, you know, yeah. Let's see here. Centromeres, they're the what do you call it? Sister chromatids. These two are attached at the region with many DNA repeats, or the center, basically. Sister chromatids, they're identical DNA strands that remain attached until mitosis. So I guess these would be sister chromatids before they detach. Let's see here. Homologous chromosomes. Okay, here's the better definition. A pair of chromosomes with identical features. So one from mom, one from dad. That makes sense. It's like, you know, if let's pull up what's it called? Karyotype, yeah. So each of these different ones are homologous chromosomes. So these go together, you know, those go together. And then X and Y, because it's an X and Y, you can tell it's a male, XX is female. Do not forget that. How long is this song? 30-minute one. I'm skipping it. It's the 30-minute one. All right. Let's see. I lost my place. Okay. Diploids and haploids. So there's there's 2 chromosomes, right? 2N. And then so it's the number of average chromosomes in a somatic cell. So there's 2 chromosomes from each of each kind. And then you have haploids, which is just N, you know, cells that only contain 1 of each chromosome. So here, I'll give you a visual example. No, you can ignore the picture, where the picture doesn't matter anymore. So like this, see? So diploids, there's the pairs of homologous chromosomes, but then haploids, there's only 1. And then you'll see those in gametes, or sex cells. Alright, let's see here. The cell cycle, let's pull up. Get the most detailed picture so I don't have to keep changing. We'll just start with this for now. Okay, so there's 4 distinct phases. You have G1, all of this is interphase, but you have G1, S, G2, and then you have the mitotic phase. There's also the meiotic phase, but that's a little different. So interphase, it's a majority, as you can see, it's a majority of the cell's life, and then this is where the cell functions as normal. But then each of these different phases you'll see here, like G1, cell growth, DS phase, DNA synthesis, G2, cell growth again. But I'll go into the specifics right here. So G1 is where you recover from the previous division and where you start to grow in size. And then so you increase the number of organelles within your cell. The chromosome is not condensed yet, the chromatin. So yeah. And then S is where replication occurs. So this is where the DNA copies itself. And then, but before S phase, the chromosomes only have 1 chromatid. And then after S phase, chromosomes have 2 chromatid. If that makes sense, if you remember what those are. So like, this is before, and then this is after, if that makes sense. And the chromosome is not condensed yet. Chromosome does not condense during interphase. But then during G2, cell synthesizes proteins to assist in cell division, so it's getting ready for cell division. And then the way they know like when to move on, it's cell signaling again. So, and then there's also a G0. Let me see if another one of these graphs, right here, yeah. So it's basically where the cell, it's where the cell just continues to function and doesn't divide. And these are usually for specialized cells. So for example, like nerve cells, muscle cells. And so it controls cell division where basically there isn't any. All right, now we're on to the mitotic, or is it meiotic or meiotic? I wrote meiotic proteins. So let me, here. Is it gonna show them? I might just look one up for each. Okay, so if you remember what the centrosome, oh, actually I haven't explained it yet. Okay, so here are centrosomes, right? So you have, within the centrosome, you have microtubules, and you have centrioles. I think that's how it's pronounced. And then each of them, there's 2 centrioles and a bunch of proteins, and they're located next to the nucleus, and they aid in cell division. The centriole itself, it's a barrel-shaped organelle. I don't think any of these are all centrosomes. Centrioles. No, these are. Okay, so these are the centrioles, right? And then so they're barrel-shaped, and they organize the meiotic spindle, which are, here we'll show, are these this? Hold on. Okay, so this is what the meiotic spindle looks like. It's these. Is it? Yeah, it is. So it helps line up. It helps. I think it's either to line up and pull apart the sister chromatids. Sister chromatids, or it's just the splitting them apart? I forget. I don't think that's too important. Then, let's see. They're located at a right angle. I wonder why, but I wrote that in my notes, so it must be important. Let's see. Then you have the spindle fibers, aka the microtubules. So these spindle fibers, same thing. And they're responsible for moving... Oh, so these are responsible for moving the chromosomes around during mitosis. And then the connectochore? Regions of the centromere or spindle fibers will attach. So if you remember, the centromere is that center part of the chromosome. So, like, for example, it's where my hands are meeting. That's where the centromere would be. Alright. So, M phase. Now we're into mitosis. So... Alright. Here we go. This is a good... Alright. So, we have... and just know, cytokinesis, it's where... So, after telophase... it's where the cells then actually separate. You're splitting of the you're dividing the cytoplasm into so have two distinct cells, even though they are identical. Let's see. All right, prophase. So interphase is what we're all doing before. It's the G0, G1, S, and G2, and then prophase. So this is where the chromosomes condense. So they condense into, you see they're uncondensed here, but then they condense here. And then so the spindle fibers form, which will assist in moving all the chromosomes to the right place. Chromosomes composed of duplicated chromatids. Yeah, and then the nucleus is gone, I think, at this point. The nuclear membrane also starts to break down, which would then, because then it prepares for duplication. Goofy song. We're moving on. Yes. Okay. And the nuclear chromosomes are not oriented yet. That will be in metaphase. And then you have prometaphase, which is like late prophase, according to this graph. And then, so this is where the connectomeres or connectochores appear. If you remember what the connectochores were. They're the regions where the spindle fibers were attached, so they're start to appear on the chromosomes. And then the spindle fibers attach to those connectochores. But they're not aligned yet. That's metaphase. So you can think of it as PMAT-C? I don't know. It's PMAT. And then metaphase. So this is where the chromosomes are aligned along the middle of the cell, like you see here. Anaphase is where the cytochromatids are separated. The spindle fibers shorten and they pull the chromatids to the poles of the cells. And this is the shortest phase, apparently. And then I'll show you an example here once we finish talking about this. Then you have telophase. The spindle fibers disassemble. They're no longer needed. And the nuclear envelope starts to form around the chromosomes. And then the chromosomes then start to unwind. And then cytokinesis is the division of the cytoplasm. But then there are some errors that can occur. So if you have mitosis without cytokinesis, then you'll have a cell with two nuclei. But if you have cytokinesis without mitosis, then you'll just have a cell with no nucleus because they weren't duplicated. Let me show you. Yeah, mitosis. Okay, so here, it's easier when it's labeled, because like for example for a quiz my teacher had certain spots labeled. But you'll see here, let's see, like for example, this one you can tell that this is anaphase, because you can see all of them being pulled apart. This is metaphase, because they're all, they appear to be aligned in the middle. And then this one, you see the chromosome is, I think this is condensed. No, you can see because you can tell it's all in the nucleus. So it's uncondensed. So this is interphase. And then this is when it, I think, is condensed because there's no nuclear envelope. And then so this is prophase. I can't say for sure. Yeah, see, because then within a cell, then telophase, anaphase, metaphase. Why do they switch these two? Prophase. It's backwards. Prophase. Yeah, what the freak? Okay. I'm gonna move on. We're still not back to these notes yet. As a result of mitosis, you have two genetically identical diploid cells, and then typically only 5% of cells are going through mitosis at any time. Think about if you were replicating all the time, that would, I don't think that's necessarily the best thing. Cycle cycle regulation. Alright, so that's what I was talking about. It's this guy. Okay, so this is a good picture. We'll talk about the cycling. So, growth factors, there are signals received by the proteins in the plasma membrane to divide. So you'll see here. And then, so cyclins are internal signaling proteins that increase or decrease the amount of cellular division that occurs, and there's hormone types, which are all here. You have, they are not all here. Where are they? Okay, here it is. Oh, so they just have different names. So the G1S cyclin, this is the D cyclin, so this is DEAB. DEAB, that's what it is. I'll just put this one up here for the sake of my notes. So cyclin D triggers the move from G0 to G1, and then also from G1 to S1. You can see that cyclin D is pretty present in these two, but then decreases as mitosis begins. And then you have cyclin E, which it prepares the cell for DNA replication, so you can see it spike right before S phase. And then you have cyclin A, which activates DNA replication. So as you can also see, it increases in S phase and then decreases afterwards, even though it peaks in G1 phase for some reason. And then you have cyclin B, which promotes the assembly of meiotic spindle and other tasks in the cytoplasm, which makes sense because you need those for mitosis. And then so you'll see it peaks in mitosis, in the mitosis stage. CDK, let's see, is it here? I'm pretty sure it was here. Yeah, cyclin-dependent kinases, or kinases. And so these enzymes are always present in the cell. There was motion detected at my front door. There was motion detected at the front door. But yeah, so there's... so enzymes are always present in the cell. You'll see right there, CDK. And then they need to activate via cyclin proteins, and then they initiate different aspects of the cell cycle, I guess, depending on the cyclin protein. M cyclins, they send CDKs to the M phase, targets proteins that make spindle fibers from, blah, 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 blah. Do we actually need to know that? I don't think so. Alright, now we have checkpoints. So you can see we have one of the checkpoints here. Yeah, here, this one shows all the checkpoints. So G1 checkpoint, it checks to make sure that the cell has enough room and organelles to replicate. G2, it checks that the DNA replication has finished, and then also checks for DNA damage. And then you have the meiotic checkpoint, which just checks if the chromosomes have attached to the meiotic spindle correctly. Let's see, and then there's a specific one, P53. Okay, so P53, and we'll talk about its dysfunctionality in a little bit. We're almost there. It's basically, so P53 tries to initiate, so if it stops it stops cell division if the DNA is found to be damaged. And then so what it'll first do is it'll try to repair the DNA. So you can see here, it'll try to repair the DNA, but if it can't, then let's see. Yeah, if it can't fix it because there's a large amount of mutations, it'll initiate apoptosis. And then it's in the name apoptosis. It's cell death, basically. And if the P53 is functional, then it stops the spread of bad mutated DNA. But if it's non-functional, the damaged DNA are passed on, and then they what do you call it? And then they just continue to keep multiplying and then keep spreading these cells. Yeah, sorry. Could read that more elegantly next time. See, when a cell becomes cancerous, body will normally... Okay, so now we're talking about cancer. So when the cell is cancerous, the body will normally go through apoptosis. So actually, getting cancer cells is more common than you think, probably. You do get cancer cells. It's just our body can usually respond and initiate apoptosis before it spreads. But then sometimes it can't. But it uses these. So let's see if it brings it up. Okay. This was an AP Bio thingy. I do remember this one. Okay, but basically, what caspasis is, they're enzymes that are usually inhibited, but they break down internal and external structures. So if the cell loses contact with neighboring cells, nucleus fragments and membrane develop blisters and get broken down. That's what I wrote. I have no idea what that means. Good luck with getting a 5. I had to work. I've had to work so much I haven't got to study. I'm hoping for a fat 3 or 4. I believe. I think it's. I don't want to say it's a glorified vocab test because that's more like AP Psych, but it's what do you call it? Hold on. What am I thinking about? What was the glorified? No, that is psychology. Psychology is basically a glorified, what do you call it? Vocab quiz. I wish I could take it, but I can't. It's in par with my graduation, and then I'm not here during the late exam, so that sucks. But besides that, it is, pretty good, pretty excited. But we're just grinding right now, and that's what we're doing. Thank you for the good luck, though. Let's see here. Cancer is uncontrolled cell growth. So now I'll pull up an example of cancer. So like you'll see, these are the mutated cells, cancer. And then there's proto-oncogens, genes that code for proteins that promote the cell cycle and prevent apoptosis. Becomes oncogens when mutated. Tumor suppressor genes, they're genes that code for proteins that inhibit its cell cycle and promote apoptosis. So that's to help prevent cancer, but then when they're mutated, they don't function properly, and therefore they don't properly promote for apoptosis. And then now we have carcinogens. You've probably heard that word before. They're cancer-causing substances that can lead to DNA mutation. That's essentially what cancer is. And then so there's, as you know, there's radiation, there's smoking, there's unhealthy drinking and eating, and there's environmental things. So UV rays, if you don't know, the sun, you are always, whenever you go outside, you are exposed to radiation. And that's because the UV rays itself is technically radiation. UV rays. And then also tanning lamps and x-rays. This is why you shouldn't do tanning. You shouldn't go to those tanning places. I think they're incredibly unsafe. Let's see, smoking, unhealthy chemicals in the cigarettes and tobacco, unhealthy drinking, alcoholic beverages, processed foods, and the environmental, like the car exhaust, fumes, and work chemicals. Alright, this is a tumor. Tumors are cancerous masses of cells, and then there's two kinds. There's benign, a cancer that doesn't spread, but then there's malignant. If you can get the similar roots, it sounds like a malicious. And then there are cancerous cells that spread. And then there's metastasis. That's where cancer cells move around, or metastasis, I think that's how you pronounce it. It's when the cancer cells move around the body, and so therefore they spread, like you can see right here. Let's see, is there a definition for that? Yes, here it is. Anginogenesis? I think that's how it's pronounced. But it's basically, it's when the cancer cells, they form blood vessels directly into the tumor, which gives them more resources and allows them to spread. Let's see. Cancer cells lack differentiation, eventually leads to the loss of function of organ and tissue. Makes sense. And then so cancers are 99.9% similar to normal cells, which make it difficult to target cancer cells versus normal cells, if you're wondering why it's such an arduous process to get rid of them. And then, but there are types of therapy. There's chemotherapy, so it's treatment that uses powerful chemicals to kill the fast-growing cells in the body, but then they also do kill good cells, which is why chemotherapy patients don't have hair. There's also surgery, where you can just remove the tumor. I think that's also given that it doesn't spread. I think they usually do this with benign. There's radiation therapy. It's where you use radiation to kill the cancer cells. And then the problem is, though, it has high side effects and low post-treatment protection because it can also kill healthy cells, similar to chemotherapy. And then there's immunotherapy, which uses biotechnology to train and act like your own immune system to fight cancer. And then part of that is antibodies, which is medication that targets cancer. All right, that's all of mitosis, and then also, like, you know, cell. I don't think we've learned about organelles yet. Oh, No, we literally did organelles yesterday, I remember. We're almost at second semester because we're getting close to photosynthesis. I don't know if you can see, but that is a chloroplast right there. Okay, cellular energies. Tell me I have that. Oh, thank God. Okay, I've been really upset. Cellular energies is mayonnaise an enzyme. Get it? I guess he says, is mayonnaise an instrument? This is what you need to know. Just if you, for me, I just do all the notes and then I know it. All right, so let's talk about energy. I guess I did write down two things. I guess because I knew the rest of these. So energonic releases free energy. So then your G is delta G is negative. Let me refresh myself on what delta G is. AT chemistry. So Gibbs free energy law. We do learn that in here. This was awful. Delta G. So is the change in free energy. Delta H is the change in enthalpy, and then delta S is the change in entropy. I think I defined those later. It's really confusing. Exergonic reactions, they release free energy, and endergonic absorbs energy. It makes sense. Exer out, endo in. Let's see. Okay, so I did write a few more here, or actually I wrote out all of these. I don't think I need them though. Specific due to the shape can be changed. Feedback, whatever. Okay. So energy is the ability to do work. That's its official definition. And then so it allows cells to carry out the essential life processes. You'll see plants create their own energy using sunlight. And then humans, we eat other things. We consume other things to get energy. We're not talking about like what do you call it? We're not talking about conceptual energy. Not like, you know, I feel so energetic. I feel like I could do a million things. That's a little different. Not what we're talking about here. Oh, my ear's oily. No. Alright, kinetic energy is energy in motion. You probably know that. Potential energy is stored energy. So, for example, if the ball is up here, it has potential energy. And then as it begins to move down the hill, as it begins to move, that is kinetic energy. And then so you have chemical energy, that's food. And then you have mechanical energy, which is movement. Alright, so now we have some laws of thermodynamics. Let's see here. So the first law, I think I summarized it here. Energy cannot be created or destroyed. It must be changed or transferred. It's also the law of conservation of energy. It's similar to the law of conservation of mass. Mass cannot be created or destroyed. It can only be changed or transformed. And then you have the second law of thermodynamics, which is that energy cannot be changed between forms without a loss of usable energy. Because nothing is 100% efficient. That's why perpetual energy machines don't work. I'm going to say perpetual motion. Perpetual motion machines don't work because, like it said, energy is not 100% efficient. Therefore, they're impossible by the laws of nature. And then so in this case, you'll see first law. So energy cannot be destroyed. It's just transformed as it moves. And then as you move, you do lose some energy, even if it's a minuscule amount. And this is specifically due to entropy. All right, let's see. A bunch of these. Oh goodness. Oh goodness. Okay. Definitely me not getting a little tired. Let's see. Energy. Some energy's lost as heat. Some energy's lost as heat. See, that makes sense. So that's like, you know, when you eat, you feel warmer, blah, blah, blah. Metabolism is the sum of all the chemical reactions that occur in a cell. And then reactants and products, I hope you know those reactants are what react to make a product. And then, so in this case, since it's reversible, I'll explain what that means. So your reactants are these two, and then they become this product. But then this product can also become a reactant, which then break into these two products, if that makes sense. Anabolic, and then catabolic. So anabolics, they create bigger molecules from smaller molecules. And then catabolic involves breaking down of complex molecules to release that energy. I think, did I write down a mnemonic for that? And then you can assign these. So anabolic reactions, they're endergonic because you need energy. You need to take energy to make those molecules. But then catabolic is usually exergonic because then when you break down the complex molecules, you release energy. I did not write a mnemonic. What I'm thinking of right now is that cats like to break stuff, and so that's how I'll remember it. Or you can also think of, like, you know, cations and anions. Anions, they're negative ions, and then so you can think of it as like a decrease in energy because you needed to put in energy to make the product. But then cations are positive ions, and therefore when you undergo the reaction, you gain energy. Energy is released from the reaction. And then, yeah, we just talked about it. Endergonic reactions, reactions that release energy, or exergonic that release, and then endergonic that take in energy. Energy of activation, you'll see these, you'll learn about this in AB Chem. I'm just stretching. I'm also getting distracted. Just give me a little bit. Let me be distracted. Is Jacob tall now? What? I have no idea what I just looked at. Okay. But anyways, so the energy of activation, it's the energy needed for all these react with one another. It makes sense. So you see, so in order to move from reactants to products, you need like energy. You need to put in energy in order to make it happen. And then so you can also tell, depending on like what this graph looks like, if you see the energy of the reactants, since the energy of the reactants is greater than the products, so this is an exergonic reaction because it releases that energy. But then if your reactants has less energy than products, then it's an endergonic, so you need to put in more energy than is released. Your net energy release is if it's either greater than zero or less than zero. And then, so a catalyst is a substance that increases the rate of chemical reaction. So it doesn't change the reaction itself, it just helps increase the rate by decreasing the activation energy. So you can think of it like this. Enzymes, they're proteins that facilitate a chemical reaction, and they're reusable, and they are catalysts. This would be an animation. You see the subject going and break apart. And then so there's two main functions. They kind of join molecules together so they can break molecules apart. And enzymes typically end in base. So, so you see here, amylase, sucrase, isomaltase, maltase, lactase. We're talking about lactase breaks down lactose. I gotta take a picture of this. This is hilarious. Just like a heart attack. I think this is big copyright, so we're gonna skip this one. I do like this one. This guy did not make Death by Glamour. I don't know how to tell you that. Okay, there we go. Alright. Yeah. Okay, substrates. This is also an animation that's not playing. Sorry, because I turned these on the PDFs. So substrate is the reactants of enzymic reactions. So they're the stuff that go into the enzymes. And then you have the active side. They're basically locations on the enzyme that come into direct contact with the substrate. Is where the substrates go. The enzyme substrate complex is the intermediate when the substrate is bonded in the active site, and then the product is what is produced, which is different from the substrate. Makes sense. And then you'll see the enzymes are 3D, I guess you know, and the enzymes are specific to certain substrates, and then based on the structure of enzymes and substrates. Hold on. This is getting really annoying. I'm turning off notifications for a second. Yeah. Induced fit. So when a substrate initially binds with an enzyme, it might not be a perfect match, but then both the substrate and enzyme change shape. So it's like, I guess you could think of it as like readjusting. Like maybe you meet someone you really like, and then you both change your personalities to better fit each other. That's the best example I can think of. Factors that influence reaction rate. The maximum reaction rate of an enzymic reaction when an enzyme is saturated with a substrate. So you can think of it like this. You can have a lot of substrate, and then so you can have a lot of enzymes, and then you can have a lot of substrate. But at some point, it can only be so efficient. I think that's what it is. Yeah, we're still here, right? So substrate concentration. So at some point, if you have, let's say if your enzyme is your limiting factor, so you have the same amount of enzymes, and then you keep adding substrate, you'll get more product, but then eventually, you'll just stop because the enzyme can only act so fast and break down, And then so this is stuff that can affect it, similar to denaturing. So as temperature increases to a certain point, the reaction rate increases because if you think about it, molecules, when there's at a higher temperature, their energy increases, and then therefore also it increases the amount they move around. There's a law for that. It's 1/2 mv squared, which is the law of, I think, the equation of kinetic energy. Let me double check. Yeah, translational kinetic energy, this. Oh, actually, let me explain this real quick. So basically you see, if you increase mass, then the kinetic energy increases if the velocity was the same. If you increase velocity, the kinetic energy increases. But then you can also use this to compare. So if like the kinetic energy for one versus the kinetic energy of another, and let's say they're equal to each other, right? So they have the same kinetic energy. You can infer that if these two molecules have the same kinetic energy and one of them is heavier, that means it must be slower than the other one, but the other one must also be lighter. This is agreed. Oh, right. Part of my talking about is to be really personalized. Like you can hear these are unique vocals, but then nowadays most of them just use the sound font, which makes sense. It makes it a lot easier to get out, but it feels less cool. Factors that optimal pH. The pH can regulate structural configuration, aka if it's outside of its optimal range in nature. Most humans work at a pH of 6 to 8. I believe this is one of the enzymes within our stomach, stomach acid. Factors that influence, we have more. So denatured is what I was just talking about. It's when bonds break, if the temperature pH rises past a certain point. So then, as we talked about with folding, it basically it unfolds back to its primary structure. I don't think it goes to a secondary structure because those need H bonds. Those use H bonds to maintain the shape. So yeah, enzyme inhibition. Right. So we're almost done with this one. Enzyme inhibition. So it occurs when a molecule binds to an enzyme to decrease its activity, and there's two kinds. You see competitive inhibitor that bond directly to the active site, which prevent a substrate from bonding because there's nobody there. But then the non-competitive inhibitor bonds to the allosteric site, which is somewhere else on the enzyme, and changes its shape. So then, the substrate can't bond because the shape is different and it doesn't fit. That's what I just talked about. Enzymes usually not apparently. So I guess allosterics can, because if you think about it, competitive inhibitors, when the inhibitor bonds to it, it doesn't itself change the shape. It just takes up the space that would be bonded, the substrate would bond to. But the allosteric site does actually change the shape. Non-competitive are, allosteric site, and the non-competitive inhibition works with the allosteric site. Allosteric activation, so that's the opposite. So when when the binding of one ligand enhances the attraction between substrate and the active site. Did I write an example for that? So it's just the opposite of inhibition, basically. Enzymic pathways is the series of link reactions. So for example, you go from here to get to here, to get to here, to get to here, to get to here, to get to here. That's what the link is. And then so this is the pathway. You go from here, and then I guess then you add these together. So these two, they combine, and then these two combine to make that, and then these combine with that, and then you get that. Melanin. Those were melatonins. Enzyme cycle, so enzymic pathway that doesn't have a specific end product. Is there an example? Let's see here. Alright, so a misconception is that the cycle doesn't go around in a circle. So they can just happen spontaneously. It's similar to the light dependent, the Calvin cycle. It doesn't all happen just like in this circle. It's just easier to represent that way because you have a start, you have a quote unquote, you can look at a start, you can go to the end, but they just kind of spontaneously happen. And then Compartmentalization. So basically, that's also what eukaryotic cells have, because then you're able to have certain reactions occur in certain places, which makes it more efficient. Rather than, so think of it, it's like a neat desk, rather than just having everything like spit all over there. So here, compartmentalization is the division of something specific, something into specific sections for functions. It's like our house. Low concentration, high concentration, high concentration. Can you tell why? So within the space it's in, there's a low amount of these symbols based on compared to the relative environment. For high concentration, there's a high amount per the environment, but then this one, even though it has the same number of symbols in both of these, the amount of space that it takes up in its environment is still very high. Smaller spaces within a Warhol. Optimizes efficiency of processes. You can see here eukaryotic cells. Prokaryotes don't have membrane-bound organelles, but they still compartmentalize in different regions. So I guess you can see like genetic materials here, ribosomes, blah blah blah. Feedback inhibition, we already talked about this, but let's see what it says here. Product produced by an enzyme binds to an enzyme active site. When the product is abundant, product forms with the allosteric site. There was a word for it, but we'll talk about it later. There are, it's not inhibitors, what's the word? It's like inducible. We'll talk about inducible reactions later. And it's basically saying, like, for example, let me find it. we get to it, I'll let you know. So this is an example. Metabolic pathway disease. So mutation occurs on genes that create one of the enzymes. So then you can't make the end product. So, redox reactions. Okay. This is the last thing, and then we're done. This is different. Or actually, this is what we started the year on. No, we did photosynthesis first. But redox. So you've probably heard about these in biology, or no, chemistry. So you can think of it as oil rig. So oxidation is losing electrons, and reduction is gaining electrons. So basically it's the and then you'll see these in certain reactions. I didn't write much notes here. See, oil rig, adenosine triphosphate, cells main source of energy. And so through hydrolysis, if you remember, so that's the adding of water to split these two, I guess to split the bond in this case. And then so energy is released. And then how ATP performs work. Exergonic is the release of the inorganic phosphate used to do, and then it's used to do endergonic work of the cell, which needs to take in energy. And then when ATP is hydrolyzed, it becomes ADP. What the freak is that? I'm like pushing something in my arm. That feels really weird. Alright, transport. The mechanic work. We saw that video yesterday. Alright, so that's it for this. We're going to be on to photosynthesis, and we're finally on this semester. It does appear it does look like we're about halfway now, but this calls for a break. So give me 1 moment. I didn't expect. Oh, did I pause the music? I'm sorry. I didn't expect that I would learn. Whatever. Instagram Reels is a weird place. Okay, let's see. We're gonna take like a 15-minute break, and we're just gonna do some relaxing things. So we're gonna rename this to taking a break. Should be on the top at all times. See, that's also the problem. I guess usually it's not that long, so I'll just put it right there. Oops. Alright. I haven't played this game in a long time. So we're gonna see how this goes. It appears I have a modifier on because I don't think I'm supposed to play. Oh, you've got to be kidding me. Oh, thank goodness. Just for funsies. Let's see here. Let's do increased spread. I'm sure I can deal with that. Heal at the start of every island or health pickups. Let's do health pickups. 2 times test. All enemy attacks poison you. Alright, just be careful. I don't know if these can hurt you afterwards when we're not going to test them. See, and we got 2 chests for winning. Let's see. 2 more choices on anvil, mag size increased. Let's do mag size. Chests contain more loot. Yes. No healing. Yes. Always want more chests. We're going to let these guys spawn in for a little bit. Following me, we're going to load this up. There. See what we got here. Freeze damage. Shock and damage nearby enemies every few seconds, or do I want to freeze them? Let's do freeze. And damage increased to enemies if they have exactly 1 debuff. Poison. Let's do this one. We'll do damage. Out of here. Get me out of here. Maybe don't go right in front of the shotgun. Okie dokie. Let's see here. Time explosion. Abilities deal dealt over time. Shots deal more damage for each bullet. Shots deal more damage when hip firing, but less when aiming. Shots deal more damage, but fire rate is reduced. That's not awful. Oh. 25% in base health. I like luck. I love that. I love that memory. It's so satisfying. No. Not what I want to do. Also not what I want to do. To enemies, but there's a short delay. Nothing. Cover for my damage instantaneous. It's also fire music. Let's see. About 5 minutes. We've already got so much done. Let's see. What luck stuff do we get? Large enemy, small chance to drop loot. Destroying has a small chance due to a higher rarity. I do a lot of destruction of that, so let's do those. Didn't need to pop those. We go in those and then get more health. Let's spend the money first. Enemies at closer range, drop more crystals, max health. Max health multiplier increases 3%. Let's see here. Heal at the start of each island. Shoot enemies. Abilities exploded with damaging bubbles. And we'll do this one. But we got one max health. Not worth it though. Alright. This is gonna be difficult. Not what I meant to do, but that's okay. I wanted to use it anyway. Okay, let's take some poison damage. Oh. So why you're moving at a Mach 10. Oh, I'm about to die. I actually might lose. I didn't realize how low it was. I'm gonna try one more time, and then we're going. Oh, falling barrels. All right, and afterwards we're gonna resume our biology studies. Alright. Let's see what we get here. For funsies. Ooh. That's good. Especially early on. Health rapidly regenerates with the first 60 seconds on that island. Heal when eliminating enemies within 3 seconds of taking melee damage due to the generator. All enemies have icy attacks. Alright. Not what I meant to do, but that works too. See? But then you get trigger, you just have to deal with hard enemies. Like those slugs. Just gonna focus on not dying right now. And watch this juke. Just take care of that rub. Okay. I didn't like that. Exposes enemies do more damage than they create. Okay. Ooh, hold on. I'm gonna try to get that. I'm pretty sure they disappear after you die, so... or after you kill the enemies, so let's not do that. Okay, trying again. But really quickly, because... There we go. See? Iron Claws. Melee cooldowns are also increased. Let's test it. Yeah. Alright. So upgrade. Hope we get the... Yeah. There we go. Let's do this one. And we're going to do luck. I just got a notification. One of my friends sent me a reel. Oh, you really have to do that. Those? Yeah, that's what I was wondering. Yeah, look at that. Ooh. That's a lot of damage. Right. That's a lot of damage. Let's see here. Streamer loot. Destroying. Let's do... Loot drops have a chance to be upgraded to a higher rarity. Let's do that. Spike chest. Since I believe we're about to go to one of those islands. Gotta be kidding me. styling on them. Right. So max health. Okay, that is a little problem. Let's see. Let's see, how much damage have we increased by? 8%. Let's see, chance of firework. Damage increased by 1% for the 2 crystals you have. I think this is better for me. For more and more loot. Healing. We'll do that. We got this. Oh, what the freak is that? Extracts. Alright. Let's take care of the more dangerous enemies. These guys. Oh, and you. Close, which is why I do that. So we finish this gauntlet. Another rarity pickup, 1 crystal bundle, armor. Let's see what we're doing here. You are going to be really annoying. Oh, that's not what I meant to do. That was a waste. That's okay. Oh, and also these things stack. Good to know. Get up, get up. Oh, we're regenerating. That's not cool. This is enticing enough, I'll do the next one. But epic pickup. Homing barrels. Let's be safe. Crystal, armor, and armor. Dashing reloads your weapon. So let's test that. Oh, it's like an instant reload. That's awesome. It's been 17 minutes. So we just got 3 crystals, basically. Abilities deal much more damage. Abilities stick to enemies and deal more damage without a delayed explosion. Shots deal more damage, movement spiral pattern. Shots deal more damage dealt over time. Let's see what else they have. Max health increase after increasing each island. Max health increased. Shots deal more damage with recoils increased. Destroying has a chance to spawn loot. Shot prices are discounted. There's a huge increase in health. see, can I? Yeah. How much damage is that? Whoa. Okay. Just need another melee strike. So, oh, not like that. This is actually so much easier than the other ones. I think just instantly play the other one. Yeah. There we go. So we can get health. Let's do... Oh. Have to pay to unlock these slots. Interesting. Let's increase our max health. That is a huge increase. Get a key. Epic. Let's see. Arcanifies. Let's see. 3% after 25. I think so this one, if I did it would be 4%, but let's do big chests. Or actually, because I can't afford any perks. Do a melee one. New region. This game has updated a lot since I last played. For the better. Oh, actually, I don't want that. So, yeah, because I don't like the zigzag, so we're going to salvage that. Let's see here. Big shots, slower, damage increases the energy. Shots do more damage, but heavy spread is fine. Negative damage. What's this total? Speedy Gonzalez. Nice. Oh, I kind of don't like that sliding sound. Let's see, shotgun blast. Oh, this is streamer loot. Chance of a deadly shotgun blast from a shot. I do like that. Let's do spike chest. Checklist. Chest contain more loot. We're sending it. We're going for another one. I feel like I'm doing a melee build instead of like an actual gun build. Nice. Ooh, damage. I know a few perks that would go really well with that one. If I want to, any players that do not have an enchantable piece of loot will still get a random chest. I wonder what enchanting a relic would do. Unless you got it. Alright, ready? no healing. See the other one. I wonder if there's anything on top of there. The first step would be to look for something like a launch. Nice, and keep raining. Alright. Next one. Heavy crab. Nice. My guess is there's probably a secret chest somewhere here. Should be enemies. I love that. Great energy. I don't mind the additional cooldown. That's fine. What's the next potential reward? Random rarity pickup. Energy rings. Okay, one eliminated, so... most. Okay. See, time explosion. No, not all the time. Okay, armor, crystal, greed. Both outgoing and incoming multipliers are heavily increased. So this I just have to be careful. Poisonous armor. Poison nearby enemies when breaking enemy armor. Ultra shot, chance to higher. Alright. This one. Let's see. Maybe. Let's take the shot first. Shots do more damage. You know, ammo's used for a short time. Checklist. If I sell that with that, well, that'll be plenty. Let's sell something cheaper. be out of bounds. Interesting. Watch this. Never finish it. That was tough. Clearing each island. Shockwave. Shockwave blast. Ski drill. Landmines rain from the sky, random. Arcane because I have that. Let's do damage. More damage and fly back towards the spawn point after a short delay. Just do more damage. Shot and nail, not missing. We'll do skill chest. Explosion. Greater radius. Let's see. Shots to knock back enemies, causing them to take knockback damage. Alright. Do damage. Scaring me. Not even did last. Fire rate increased. Okay, let's see. Arcane Blast, Fireworks, and Explosion Rings, bonus crystals, valued customer. Let's see here. Piercing shot. I think I like the piercing shot. And yeah, we'll see how it goes. There's the chance we'll roll one more time that it might get armor. Don't know. Dagger arc. Chance to fire daggers. Okay, 2,000. 7 of these perhaps. Let's see. This be enough. Okay, just one more. Let's see here. Oh, I get it. Okay. I think it was really close to winning too, but that's okay. That was a good run. I got a bug bite apparently. Okie dokie. We are back to locking in. So we're gonna turn the music back on. Now we're going to be talking about photosynthesis. Here we go. You can barely see it, but it says Unit 3 Cellular Energetics, the photosynthesis. Alright. Okay, so to start, autotrophs are organisms that produce their own food, hence the name auto. We already talked about this. Heterotrophs, you need to consume, it's what we do. So, at its core, this is what photosynthesis is. 6 carbon dioxides plus 6 water, and then go to, with light energy, they become sugar and oxygen. But then we'll get into the more specifics in a second. So we have the stomata, which is it's different from the stroma. Make sure you know that the stomata is the opening in the leaves of plants, which allows the CO2 to enter. And then water and oxygen leave through this way, so like perspiration. The stomata is plural, but it's different from the stroma. Right. So let's see. Light intensity. So I guess it can only work so quickly. So at some point, if you add a lot of light, it's just not going to increase the rate. If you add a lot of carbon dioxide, again, it's not going to increase the rate because you need to yeah. And then I guess temperature, there's an optimal temperature, which makes sense. Saturation, that's what it is. Increase the point of saturation, where it's the excess. Average weight of a redwood is about 50,000 pounds or 25 tons. Why isn't there a large hole around the tree? What? Why would there be a large hole around the tree? Majority of mass comes from CO2 in the atmosphere, non-nutrients out of the ground. I could use a light snack. Get it? Alright. Photosynthesis, and then so the water is split, and then so it transfers the protons or the hydrogen ions to the sugar. So you can see. So basically, if you think about it, so oxygen, I'm pretty sure always has a -2, and then so since this is balanced, this is going to be the carbon is a +4 right now. But then now, since this is -2, this is -2, so this is -12. And then this is going to be +12, and then so this is carbon is 0. So it becomes reduced because it gains electrons, which lowers its value. Does that make sense? Insignificant notification. Chloroplast split H2O into hydrogen and oxygen, incorporating the elements into a sugar molecule. But this is, so that's important, sure, but this is more important. So you have the photo, which is light, then synthesis, where you take carbon dioxide and you make it into sugar. So then now we're going to go into detail. We're going to talk about the chloroplasts. So chloroplasts have two membranes because if you remember the endosymbiotic theory, so it is of both the membrane of the original bacteria it was, and then also the plasma membrane from its eukaryotic host. So let's see, chloroplasts have two membranes. The stroma, see, this is different than the stoma. This is the stroma. The stroma is the solution inside the chloroplast. The stoma is the openings, and the stomata, those are the openings on the outside that allow for carbon dioxide to enter. Alright, so you have the thylakoid lumen. So basically, so you have one lumen, is one of the stacks, and then you have a granum, which is a stack of thylakoid lumen. And then the lumen's the inguinal. So, sorry, you have the thylakoid, and then the lumen's on the inside, and then a stack of those are the granum. And so thylakoid is where it's where the photosynthesis takes place, and we'll show you. Okay, so photosystems. These are groups of pigments and electron acceptor molecules within the thylakoid membrane, and they act as solar power for the energy. And then there's two types. There's photosystem 1 and photosystem 2, and they both accept different kinds of. Well, they're more ideal in different levels of wavelengths. So we'll go over those later, though not yet. And then so for this, you also have to know the visible light spectrum. It's the basically the white light is separated into different levels of energy. Light is energy. And then so pigments are molecules that absorb certain wavelengths of light, and then so by absorbing them, they reflect the other wavelengths, and thus those are the colors we perceive. So because we see plants as green, that means they reflect the green wavelength and absorb the other ones, which we will talk about which ones specifically later. As you can see, it's most of the other light. Chlorophyll, and then so these are found in photosystems, and they absorb all light except green, and then that's why plants look green. And then other pigments, so there's carotenoids, anthocyanins, that was on our quiz. I don't know if it'll actually be on the test. Alright, so you can see here, there's high absorbance for these colors, low absorbance for these, and then moderate here. Absorption spectrum is light that is absorbed by a molecule, so it's this one, not this one. Right. So in light-dependent reactions, if you couldn't tell, this is what depends on the light. Therefore, this is it's the light reactions, and they occur on the thylakoid membrane, and they require light. So and then there's the Calvin cycle, which is light independent, and then occurs inside the stroma, and then this is where the enzymes reduce carbon dioxide to the carbohydrates, the sugars. And it requires ATP, NAD, NADPH, and CO2. And where do they come from? They come from the light reactions, which produces ATP and NADPH from ADP and NADP+, which we'll go over more specifically very soon. But first, I guess, what? Why are they not wearing clothes? Whatever. Okay, so NADP+. NADP+ is a molecule that accepts and delivers 2 electrons and 1 hydrogen. So you can think, because you add a hydrogen, right? If you add a hydrogen ion, it's positive, so then it'll make it NADPH++, or yeah, I think it's 2+, and then so then it accepts 2 electrons, which then make it it's +2. It's 2+ plus 2 minus, which then becomes 0, because as you can see, NADPH does not have a charge. And then the ATP cycle, we've looked at this before. So ATP is charged, and then so by adding of water hydrolysis, it breaks off one of the inorganic phosphate groups, which releases a lot of energy. And then through dehydration synthesis, and then a lot of energy, then the ADP is recharged. This is, I guess, what it looks like if you wanted a more technical graph. And then there are 2 types of photosystems in the thylakoid membrane. There's photosystems 2. Okay, so it's important that you know the order that they go in. First, it's photosystems 2, and then it's photosystems 1. They're only named that because that's the order they were discovered. So photosystems 2 functions first, and it's best at absorbing wavelengths of 680 nanometers. And then the reaction center of chlorophyll A, so it's called PS 680, can you guess? Because it's the nanometers. And then photosystem 1 is best at absorbing 700 nanometer wavelengths. And then so the reaction center of chlorophyll A is called P700. P680 and P67 are nearly identical, but their associations with different proteins result in different light absorbing properties. I just read off the slides for that one because I don't think you actually need to know that specific. Okay, so alright, so this is where it gets complicated slightly. So this is the light reaction. So this is just light reaction, and then we'll talk about the linear electron flow in a little bit. Okay, so let's start. So chlorophyll is excited, not emotionally, is excited by light absorption, right? Because it's a pigment, and so it absorbs the wavelengths. And then so the electrons are passed to a reaction center of the photosystem 2. So you can think of it like this. It goes down, and it goes up, and then it goes back down, and it goes up. That represents the energy, and then I'll be able to show it better because I didn't actually write it down, but that's okay, because I know how it works. And then so, energy is passed to the reaction center of photosystem 2. I thought that was weird. And then the electron is captured by the primary electron acceptor, and so a redox reaction happens where the electron is transferred, because remember, oxidizing is losing, and then reduction is gaining. And then so the electron is prevented from losing energy, so it drops back to ground state, and then I'll show you what that means later. Hydrogen, water is split to replace the E, and so oxygen is formed. And then the electron is passed to photosystems 1 via the electronic transport chain, which we'll look at too. And then so the energy transfer pumps the proton into the thylakoid space, ATP produced by photophosphorylation, and then the electron moves from the photosystem 1 electron acceptor to the second electron transport chain. NADP+ is reduced to NADPH because it gains. And then yeah, so the linear electron flow, it just shows the flow of electrons through the photosystems and other molecules. And then so if you want to read it in words, there'll be a picture as we saw before, but we'll just start here. Photon hits a pigment, and its energy is passed among the pigment molecules until it excites the chlorophyll A. And then the excited electron from there is transferred to the primary electron acceptor, and then we call it that plus because with the electron, without the electron, it's minus. And then so it's because the electrons are negative, and so without that, it has a positive charge now. Water is split by enzymes, and the electrons are transferred from the hydrogen atoms to the P680, reducing it back to P680 or P680 plus, reducing it to P680, and then O2 is released as a byproduct. Each electron falls on an electron transport chain from the primary electron acceptor of PS2. Energy released fall. Okay, so this is really boring to read, right? It's a little hard, so we'll explain it here. So you see these are the pigments, right? And then so with light they're excited, and then they promote this electron, and the electron gains energy. And then to replenish that energy, the water is split lysis, and then so electrons are then given because it's from the protons, half oxygen. The electrons are then returned to the P680. But then, as you see, then that energy is used as it goes down the electron, we're talking about energy, goes down the electron transport chain, and then it helps produce ATP. And then it goes down to the first photosystem 1, and then, more light can excite these, so then the energy goes to the P700, and then from there, then the electron is, again, promoted, and then it goes back down the electron transport chain to reduce NADP plus to NADPH. So reduce? Yeah, reduce. And so water splits at the photosystem 2. It's called photolysis, light, and then lysis is in like cutting. And then so water donates its electrons, but then the oxygen, got a good grade. Oxygen atoms left over become O2, and then the hydrogen ions are in the lumen. So that's inside. So that's within the thylakoid. Light is absorbed by pigments in PS2. Energy transfer. Okay. I mean, that's literally what we're talking about. You can see here. So water's split, O2 and H+, and then so there's protons. There's just a ton of protons in here. This is important for later. And then this is the electron is excited, and then it goes down the electron transport chain, which allows for the diffusion of protons inside, and then which will help ATP eventually. And the photosystems 1, P700, electrons excited, and then it helps to reduce NADP plus to NADPH. This is just explaining why it's called a proton. Alright, so electrons move down the ETC. We've just explained that. PS1 accepts electrons. Step 4 is probably behind this. That's fine. Electrons pass to the membrane protein NAD reductase. It sounds like it's reductase. It's an enzyme that reduces NADP+. Right there. And so this is just what it looks like. And then so you'll notice we haven't yet talked about the ATP synthase. So what that does is so we have oxygen, right? And then so it's split photolysis to have half of O2 or just O, and then 2 H+. These are protons. And then so as the electron moves down the first electron transport chain, it allows for these protons to be pumped into the lumen. And then this is re-excited and it reduces NADP plus to NADPH. But then you'll see that now we have a concentration gradient, right? We have a concentration. There's more protons inside the lumen than outside. And then so what we do from there is then with ATP synthase, those protons diffuse out, and then using mechanical energy, because it turns a cog in here, I forget what it is, then it helps to create NADP, it helps to create ATP from NADP and an inorganic phosphate. See, we're about to talk about that here. So lumen acts as a reservoir for hydrogen ions. Hydrogen ions move through the ATP synthase. ATP synthase turns bonding ADP and a phosphate group to produce ATP. So turns, there should be a comma here to make it more obvious. These are animations. You really don't need to know. They're super confusing, but yeah. I really wonder what this one is. Alright. Chemiosmosis is a movement of ions down a chemical gradient. So in this case, this is what this is. So you have, what do you call it? Because when you pump against the concentration gradient, when you have, not facilitated diffusion, what's it called? I think it's called active diffusion. Yeah, active transport. So active transport, because you're going from a low concentration to a high concentration. But then this is just facilitated diffusion because you're going from a high concentration back to a low concentration. Or, yeah, no, I'm assuming because it says low H+, but I'm pretty sure you can't just, it'll just happen. And then, so the enzyme catalyzes the reaction, and then it creates that ATP. So then this is just a summary of all that's happening. I think this was also a video. Okay, now we're on the light-independent reactions, and then so these occur after the light reaction. How long was this one? 19. Okay. Where is... Hold on. There's... Denmark. Where's Denmark?
I've arranged for you to meet my
There. This is some old sequence music that I really love. Interesting. Okay, but so this is basically what the Calvin cycle is. So you start with CO2, right? And then with Rubisco, this is important you know what this is. The carbon is fixed into from 3CO2 to 3 PGA. So the 3 just means 3 carbon, and then PGA. And then so this is G3P, but another way you can write it, it's PGAL. Those are both correct. Those are both the same molecule. And then so with using ATP, so using energy from both ATP and NADPH, it is reduced to G3P, and then this goes to make glucose, but then one of these goes to make glucose, but then 5 of these are recycled. So G3P is recycled and is regenerated, so then again uses more energy. I think this is missing a photo, but that's okay. And then it becomes back to RUBP, which I'm pretty sure is Rubisco. Carbon fixation, so single molecule of CO2 attaches itself to the 5 carbon molecule. Okay, so that's different. So our RUBP is different from Rubisco, but actually, so CO2 adds itself to RUBP with Rubisco. Sorry, that's what it is. And then so 6 carbon molecule then splits into 2, so there's 3 PGA. And then Rubisco, so it's the most abundant enzyme on Earth, takes carbon from CO2 and fixes it to RUBP. And then so we have carbon reduction. So you have fixation, reduction, and then regeneration. That's what's going on here. Let's see. Carbon reduction. 2 reactions take place. ATP and ADP are used. Comes from the light reactions. As you know, we produce ATP and NADPH from the light-dependent reaction. And each of the 3 PGA goes through a reduction reaction and becomes G3P. Then PGAL and G3P are the same molecule. It's important to know. It's not really important to know, you just might see both. And then you have regeneration, so one of the G3Ps goes to make glucose, but the others are then reconverted to make 3 RUBP molecules. And then ATP is required again for this step. You need to put in energy. And then I guess if you wanted to see the total number of molecules, this is what it looks like. Because then you're probably wondering at the beginning, like when we were here, it's like, how do we know it makes 6 of these? And then what about the other 5? But then this is where, or this is it. See, then you're like, where did all these numbers come from? And then you see, oh, this is actually what it looks like. We're getting close to the end of this, and we're about to go to cellular respiration, which is infinitely more complicated. And then so this is with 3 CO2, but with 6 CO2, which is actually, you know, what we said at the beginning. And then so, because you need 2 of these G3Ps to make glucose. So then this is actually how much is going on at once. You put in 18 ATP and 12 NADP. So 2 G3Ps or 2 PGALs make 1 glucose, and then the whole process must occur twice for 1 glucose to be made, basically. Because you need 3 just to make 1, so you need to go through that cycle twice. And again, it doesn't go like this. It happens spontaneously. This is just for ease of understanding. And then GT3 is a starting molecule of many organic molecules, helps to produce all this sort of thing. Calvin cycle does not occur in again, like I said, these reactions just happen, and they can occur randomly. Stomata versus stroma. Don't forget, the stomata closes during hot portions. It conserves water because this is where water leaves. And then the CO2 levels drop, oxygen levels rise, obviously because they're not perspirating. Let's see. Okay, now we're in photorespiration. I love this game. Alright, photorespiration. So it occurs when O2 is taken up by rubisco instead of CO2, and it is not good. So because we lose the carbon instead of gaining it, and but it does occur when CO2 concentrations are low. So you can see here, if CO2 goes in, then we go through this cycle, but if O2 goes in, then we have all of this, and it looks vastly inefficient. See? So for CO2, we gain 6 fixed C atoms, but then for photorespiration, we actually lose 3, because you see all those, and glucose isn't made. That's the problem. Alright, so photorespiration. So when photosynthesis evolved, oxygen levels were low, which makes sense, which is why they utilize carbon. Let's see. It didn't matter if rubisco was selective for CO2 or O2. It didn't hurt the plant. Okay, so I guess that makes sense. Since there was less oxygen, and I guess it wasn't too much of a problem back then, but then now there's a ton of oxygen. Earth is pretty old. And then first life on Earth was anaerobic, which means it didn't utilize oxygen because there wasn't much. It was nitrogen, carbon dioxide, and CH4, carbon tetrahydride. Methane, okay. Cyanobacteria evolved a photosynthetic process that produced oxygen and sugar. Huge evolutionary advantage, I guess. Atmosphere increased in O2, and the anaerobic life died off because of the oxygen. Great oxidation catastrophe. CO2 is a greenhouse gas. Less CO2, what happens? Ice Age lasted for 300 million years. 2 billion years. Ozone was formed, warmed planet, protected Earth from harmful UV light. Oxygen was found in the air and water, and then so aerobic species came to dominate because now, we have oxygen, and therefore anaerobic species won't be able to utilize that, but then aerobic species can. Oxygen allowed for complex life, after. Okay. These are the different kinds of photosynthesis we're about to talk about. Yeah. So there's C3 plants, and then so these, the molecules these form are the 3 carbon. It's the one we were just talking about, like G3P, PGAL. And then, so there's no defense against photorespiration, which is what we're talking about. It's really bad. And then so it's the normal photosynthesis. It just goes directly through. And then now C4 plants. This means 4 carbon molecule. And then so these ones live in the hotter, drier climates. And then so the first thing they do. Let's see. Plants do not react with oxygen because the enzyme PEP carboxylase. So let's see. So CO2 enters, and then with PEP and then help with PEP carboxylase, they become oxalolactase, and then malate, and then I guess it's stored first, and which uses more energy, but I guess is more effective in hotter. It's like long term, I guess. This makes sense. Hotter, drier climates. And CAM plants. So, again, hot and dry climates, but I guess this is different. They take CO2 and create 4 carbon molecules which change during the day to make glucose. And so they only open the stomata during night. So it's similar. Well, I guess, you have the night. It's instead night versus day reactions. Alright, now, we're not onto this yet, but now we're onto cellular respiration. Alright, so this is what we go through as well, but then it's also whatever has a mitochondria, yeah, they go through this. I'm stretching. Hold on, we're gonna stretch. Yeah, actually, we've gotten decently far. I'll take that.
Profile new, staring nothing for an hour or two. Dropping a beat like 100 other artists would do. Clicking some chords like 100 other artists could do. Like, probably gonna hit that don't save changes again. Used to write a song a day. It's just that lately my perfection's in the way of me, and I can't make no changes to save me. Wish I could write more, careless. Wish I could write more and careless. Write more and careless and careless. Sometimes I'm angry, I'm not doing better than I thought I'd do at this point. I punish myself by depriving my health of the things that I like until I fix this. But it doesn't really help, I just get more depressed. I do even less, cause I can't work when I'm stressed. And ignoring my friends means when I'm finally done, there'll be no one left to make this all worth it. Oh, I've been thinking too much, and now 2 hours have passed, and I've got nothing to show. I guess tomorrow we'll give it another go.
Ooh, I love this one. Okay, well, we're back. I'm gonna finish this round real quick, and then we will get back into it. Okay. Look, I won. Isn't that awesome? Okay. Now we're going to get onto cellular respiration, which is aerobic respiration because it requires oxygen. There's anaerobic There's anaerobic versions of this, but they're a little different, and then so we'll talk about those. Right? Let's see here, and then so the formula. There's a. I have no idea what I'm doing here. Okay, I'm trying to understand this real quick. So ATP produced and then ATP used. Okay, so here is the general formula for a cellular respiration. Take glucose and oxygen, and you make CO2, water, and energy. That's why we breathe out carbon dioxide. Alright, electron carriers. So this is what you'll need to know. This time they're wearing clothes. That's good. So basically, NADH and FADH2 are reusable electron carriers. So what they'll do is they'll take the electrons from hydrogens, right? And then they'll make those protons. But then what they'll do is they're also reduced to NADH, as you can see. If this otherwise, it would be. See, right now it's NAD plus by itself, but now it's just NADH, or just NAD and then NADH. And then so what it does is it takes it and it moves it somewhere else, and these are reused in later stages, and then NADH goes back to get more electrons. That's basically what it is. You don't need to know the structure, that's just what it looks like, NAD, and then you have, as you'll notice, you have an additional phosphate group, NADPH. Okay, I might have to, if you guys don't know what this is for the absolute most banger. I won't show it in full because the game itself is a little violent, but basically it's for Counter-Strike Global Offensive. It is absolutely amazing. I always wanted to see what the score for this piece would be like. Maybe I'll email, or maybe I'll see if I can like take a peep. Alright, so you have the mitochondria, you know, which is, there's multiple mitochondria per cell, and they have the double membrane because, again, sorry, the song, but then they have the double membrane because of the endosymbiotic theory, and then, so the matrix is the fluid inside of the mitochondria, so inside this part, not the double membrane, that's the intermembranal space, and then you have the cristae, which are the folds of the inner membrane, which create more surface area. As you know, surface area is good. And then, so aerobic, and then anaerobic. I hope you know the difference. And there's 4 main phases of cellular respiration. So there's glycolysis, there's preparation for the Krebs cycle, and then you have the ETC. And then glycolysis is the only anaerobic phase. And then that has to do with history and evolution, and then what it can also split to. So this is glycolysis. And then if it's aerobic, so there's oxygen, then it can go into aerobic respiration. That's the preparation step, Krebs cycle, etc. But then if it's not, then what it'll do is it'll leave the mitochondria. It'll go in the cytoplasm where it goes fermentation. It'll either be lactic acid fermentation or alcohol fermentation. Alcohol is, you know, the methane, but then lactic acid, it's when you work out, it's the soreness you feel, and that's lactic acid. Okay, so notice. Respiration is endergonic because it releases energy, which is ATP, but photosynthesis is endergonic because it takes in the light energy. Hold on, give me 1 second. I heard something. What? I got food. Hold on, I'm gonna test myself. Ah! My dog is downstairs, and so I put this on the TV. We're gonna test something. I'm gonna see if there's if I can do music through my microphone. Yo, 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 yo. Okay, I don't want to wear headphones, so I'm glad that this option works. Okay, we're talking about cellular respiration. Okay, so that's the overview. Pretty basic stuff. So as you can see, glycolysis happens outside of the mitochondria. And then so pyruvate oxidation, that's the same thing as we were talking here. It's the preparation step for the citric acid cycle. Krebs cycle, same thing. There's a different name. And then this produces ATP, a very small amount in ATP, but this is the most. I believe it's 36 ATP is produced. And so we'll talk about glycolysis real quick. It occurs in almost all organisms, and it evolved before the Krebs cycle in the ETC. So it's also probably why, one, it's not in the mitochondria, but then two, it also, you can see here, it evolved when there was no oxygen in the atmosphere. Therefore, that's why it's an anaerobic thingy. And then so glycolysis, glyco, sugar, lysis, splitting, sugar splitting. It's believed to be ancient, occurs in the cytosol, so outside of the mitochondria. Partially oxidizes glucose to 2 pyruvates, so there's a net gain of 2 ATP and 2 NADH. Now, no net gain, so we'll talk about that in a little bit. It actually produces 4, but we use 2 of them. So it occurs, you'll see, it breaks down glucose, which is a 6-carbon molecule, into 2 pyruvate molecules, which are 3-carbon. And then, so here's the energy investment. So you need 2 ATPs to add a phosphate group onto the glucose, which you see, and then you have an unstable fructose. It's fructose 1,6-bisophosphate, and then so that then splits into 2 G3P molecules, or PGAL. And then so now this is where we get the energy. Each of these reactions, they each produce 2 ATP by themselves, and since there's 2 of them, they go into making the pyruvate. So we make 4 ATP, but we have the energy investment step to put on those phosphates. So we actually lose 2 ATP, so it's a net gain of 2 ATP. That's why it's a net. And then C creates 2 ATP molecules, G3P is converted to pyruvate. Yeah. And then so this is the these are the 2 phases. So you just need to know you need to invest ATP to put on those phosphates. And then since this is an unstable molecule, it breaks into 2 PGAL or 3 G3P, and then from there, each of the G3Ps undergoes the same thing. It oxidizes NADH. Actually, no. It reduces NADH because it's gaining the electrons. And then an organic phosphate. And then you makes ATP from this. And then it makes ATP. It doesn't make ATP. It puts on the inorganic phosphate it takes one of its own inorganic phosphates, and it puts it back onto the ATP basically. So it synthesizes it, let's say. And so this is a summary. This is what happens. We make 2 ATP net gain. We also gain 2 NADH and then 2 protons, and then we also make 2 water. Substrate level phosphorylation. So an enzyme passes the high energy phosphate to ADP, which results in the formation of an ATP molecule. So basically this is what happens. This is the enzyme, Alright, and then so this is where the rest of it occurs. So the glycolysis occurs out here, and then we come in to do the citric acid cycle, and then which is in the mitochondria matrix. And then we do ETC, which is in the inner membrane, which utilizes the intermembranal space and the inner membrane. So this is the pyruvate oxidation, which is the intermediate step. I'm back. I don't know why this is. So yeah, so we use the intermediate stuff. Also, I forgot that I have food, so I'm gonna eat. My food. Delicious. All these cables are making my desk messy. Just cleaning up real quick. Give me a moment. You can take in this graph already. If I was a better cook, I'd be eating more stuff. Well, alright, so now you can see here, the pyruvate comes into the mitochondrial matrix, and then it undergoes oxidation, right? So then a CO2 group is taken off, so now it's a 2-carbon molecule, and then it reduces NADH, therefore oxidizing itself. And then so we add choline, to make acetylcholine. It's a 2-carbon with a CoAA. And then the CoAA is removed, and then we have the acetyl come in and then engage in the citric acid cycle. So you can see what's happening here. The pyruvate moves to the cytoplasm, to the mitochondrial matrix, membrane proteins. We already know this is just, see how the concepts are building upon each other? I'm literally quoting my teacher here. Oh my goodness. First in the mitochondrial matrix, pyruvate is converted into acetyl, and then so NAD oxidizes the pyruvate, and it becomes NADH. So the NAD oxidizes pyruvate, but the pyruvate reduces NADH, or NAD, and then so carbon dioxide is also produced. And so this occurs twice. Wait, hold on. Wait, hold on. Coenzyme A attaches to acetyl, carries it to the Krebs cycle. Oh my goodness. So this is the acetyl, and then this is the coenzyme. That's hilarious. Okay, now we're at the citric acid cycle. Where do you think the name Krebs cycle comes from? Usually, if it has a weird name, it's either because that's the biological name, like the chemistry name, or it's because a guy had a weird name. Or let's say unique. Weird is a little rude. So this is a unique name, Krebs. I don't know what TCA stands for. It's located in mitochondrial matrix, utilizes substrate level phosphorylation because it produces ATP, and it turns twice for every glucose molecule. So we'll see here. Because for every glucose, 2 pyruvates are made, right? And therefore, 2 acetylcholines are made. And therefore, this will spin twice per glucose molecule. See? So you really need to know how these build upon each other. I really give a whole page to the Krebs cycle. Alright, so but basically here's what's happening. I don't think you need to know everything that's going on here. But in case you need to, basically here's what's happening. So the acetylcholine, it joins with the octolacerate, I think that's what it's called, octolacerate, and then it becomes citrate, citrate, which is not the 8, an acid. It has it's 6 carbon. So this itself is 4 carbon, and then citrate is 6 carbon. And then so from there, the NAD oxidizes citrate to produce NADH. But then that also creates carbon dioxide, and ketoglutarate. So we're skipping over this one, I guess. Okay. And then then NAD oxidizes this again. So it goes from 5 carbon, oxidizes, removes a carbon dioxide, and then it creates succinate. Acetylcholine. I literally just forgot what acetylcholine was. Choline. Succinylcholine. And then from there, FAD is then reduced to produce ATP and fumarate. And it's basically all I've written down. This is what if you want to read it, sure. This is basically what happens. So all you need to know is that acetylcholine joins with so this 2 carbon joins with a 4 carbon, and then from there it creates a 6 carbon, and then from that 6 carbon, NAD reduces it, or oxidizes it, sorry, and it also removes the CO2, so it becomes 5, removes the CO2, it becomes 4, and then after the 4, it then reduces, it helps create ATP, and then also, what do you call it? It reduces FAD. That's just what happens. And then also NAD again. Alright, so we had a little tally written down in here. So I'm about to read it to you. So glycolysis, what it produces: 2 pyruvate, 2 waters, 2 NADHs, and 4 ATP. But there's a net gain of 2 because we used up 2 of them. The Krebs cycle. I'll read this for 1 cycle. It creates 3 NADH, 1 FADH2, 2 CO2, and 1 ATP. But since it runs twice per glucose, really it creates 6 NADH, 2 FADH2, 4 CO2, and 2 ATP. Now I skipped the prep step. I'm sorry. In the prep step, in between, creates 2 acetylcholine, 2 NADH, and 2 CO2 per glucose. Okay, and then from there, so we've gained a total of 4 ATP, right? Like a net gain. And then so now in the electronic transport chain, holy freak, we're about to make a total of 34 ATP about. And then 10 of that comes from NAD, 30 of those come from the 10 NADH, and 4 of those come from the 2 FADH. So now we're going to talk about first chemiosmosis. We already discussed what this was before, but basically the H+ gradient drives cellular work, and the proton motive force, it uses protons. The proton gradient to perform work is similar to what we saw in the like-dependent reactions. And then ATP synthase, it's also present here. It's an enzyme used to make ATP, and it uses the energy from the proton gradient as the ions flow back across. I'm gonna take another bite, and then I'll be with you shortly. Is this also 30 minutes long? Okay, we're almost done. I'll let it finish. Two 37 views. I'm impressed. Okay. Thanks for the people who have been coming and sitting in, but now we're locking back in. Okay. So inner mitochondrial membrane, NADH and FADH2 molecules from the first step. Okay, so now they're back, right? And then so what they do is they deliver the electron, because they're electron carriers, right? They deliver the electrons to the membrane proteins, which then help with the with this electron transport chain. And we will discuss what it does specifically if I can be competent. Electrons move through the chain, it's the electron transport chain, and then each membrane protein is more electronegative, which helps move the electrons along. If you remember what electronegativity is, it's basically the ability for, in this case, an element, but these are proteins, and then it's how they It's their ability to attract electrons to itself. If you look at the periodic table, the more up and the more right you are, excluding the noble gases, the more electronegative they are. So the hydrogen ions move into the inner membrane space using the energy from the electrons. So they move through, right? These are channel ions, I believe. And so it creates this concentration gradient. And then and then we'll come back to that later. But here, at the very end of the electron transport chain, oxygen is the final acceptor, which then combines with hydrogen ions to form water. That's why water is produced. And then, so from here, as the hydrogen ions move from high to low concentrations, they do this by going through ATP synthase, which then harnesses the flow of those ions to create ATP from ADP. Because you need to put in energy in order to make to add the inorganic phosphate group back on. And so oxidative phosphorylation. So this is the production of ATP by the movement of electrons through the ETC coupled with ATP synthase. I don't think that's the same thing as what's going on in photosynthesis. Let's see. Okay, I think it's just specific to cellular respiration. We'll just play it safe and we won't generalize the term. Again, what is this supposed to be? Okay, but this is basically all of it. So the NADH and the FADH2 from the first steps comes here with the electron, and it donates it to these proteins, which are used in the part of the ETC, and which help carry the electrons through. And then that energy also allows it to pump hydrogen ions or protons into the inner membranous space. And then so it moves the electrons along, and then once it gets to the end, O2, oxygen, is the final acceptor, which then becomes H2O by bonding with the other protons in here. But then with all of the protons in the inner membranous space, note it needs energy because we're pumping against the gradient. And then so it wants to go from a high to a low concentration, and thus it does so by going through the ATP synthase, which uses its mechanical energy to create ATP from ADP and an inorganic phosphate, of course. And so oxidative phosphorylation is both the ETC and then chemiosmosis. I think that's what this is saying. All right, and then so for each NADH, 3 ATP are created. And then for each FADH, 2 ATP are created. And then so since there's 10 NADHs we've made, we'll make 30 ATP from one glucose molecule. And since we make 2 FADH2s, if you remember from the Krebs cycle, and then we'll make 2 ATP each, and so we'll have a total of 4. This is supposed to be here, and it is supposed to show the yield, but we've already discussed that. ATP yield per molecule. And so you see there's a total plus 2, and then 28 to 38, 26 to 28. Interesting. I thought it was 42, but you realistically it's not 100% efficient, so you lose some energy and some capacity. The day mitochondria went from being the powerhouse of the cell to the ATP synthesis by oxidative phosphorylation was a horrible day. Okay. Anaerobic respiration. Let's see here. If oxygen isn't present, electrons will be stuck in the ETC. Makes sense because because oxygen is the final proton acceptor, right? So you won't be able to move on. And then so NADH and FADH can drop more electrons, so really you need oxygen. This requires oxygen. And then so therefore NAD and FAD will used up, and then so everything that requires their energies will stop. And so this is where anaerobic respiration comes in. Still on this page. So fermentation is an anaerobic process that produces a limited amount of ATP in the absence of oxygen. I don't think you need to know these specifically, but lactose, cheese, ethanol, beer, wine too, I guess, carbon dioxide. So glycolysis reacts as normal. it changes NAD into NADH, but so the NAD amount will decrease, because even though the NADH has accepted protons, right, and then it's supposed to move on to the inside of the mitochondria, but without oxygen, those phases can't occur. So therefore, the total amount of NAD decreases, but the amount of NADH increases. But it is utilized in other reactions. So you can see here, pyruvate is reduced by NADH and then produces NAD that can keep glycolysis going. And so there's two kinds, main ones I think that we're talking about here. There's lactic acid, which produces 2 ATP, and it occurs when we, it's short term. The important thing to know is it's short term. And then so it's why you feel sore, like I said earlier. And then there's alcoholic fermentation, which produces ethanol as well, and it occurs in plants and bacteria, and then yeasts produce this, and so yeast will produce so much alcohol it kills the yeast, which sinks to the bottom. Interesting. Okay, now we're on triglycerides. If you think about it, what does it sound like? 3 glycerides. So they're broken down into 1 glycerol and 3 fatty acids. Glycerol can break down into G3P, and then fatty acids can be broken down into acetyl-CoA. If you know, we need acetyl-CoA, and acetyl-CoA is important for the Krebs cycle. It's what the intermediate step, the prep step, produces. And then G3P is what we need to create the pyruvate. And so here's all of that, if you want to take it in. Will I take another bite? It's really good. I'm gonna show you some thrift store finds I found. I found this bone book. I have a couple of them already. I think I'm missing 2 and 5, unless this is the last one. It is. Okay, so I'm just missing 2 and 5, and then I will have the whole bone collection. Would you also believe that I have the handbook? I don't think that's really where. It doesn't matter. I actually don't know where I got it, but I have it. Then I also got this Garfield book. My short little adventure. I love Garfield. And then I also got this. I might give it to one of my friends because I'm never playing Skyrim. But I found this. It's the whole book. It's like a whole guide on how to play Skyrim. But alright, probably taking it in. Excessive amounts of carbohydrates can result in the formation of fat. Extra acetylcholine can form fatty acids. Interesting. ATP is like an ice cream sandwich. Unless you eat it quickly, it's going to melt. ATP hydrolyzes into ADP quickly. So you make ATP from glucose, and it's not efficient to waste it. So cells don't need to be continually producing ADP. And so, this is what we call, if you remember, this is negative feed. What are we listening to? Okay, Friday Night Funkin', that's fine. This is called negative feedback, because remember, our product is inhibiting the rest of the process, so then it produces less of the product. Let's see. Pyruvate dehydrogenase. And then that breaks down pyruvate to acetylcholine. Oh, okay, so we're talking about specific inhibitors. So we know ADP activates it because, if you have ADP, that means you need more energy. Pyruvate, because the pyruvate is the reactant, and then calcium, sure. And then inhibitors are ATP, NADH, and then acetylcholine. Because we make acetylcholine, it's our product. ATP indicates that we don't need to make as much. That sort of thing. More stuff. Mitochondrial diseases. Okay, we're almost done with these slides. Okay, so mitochondrial DNA is circular, only contains 37 genes. I think that means it's... Oh, yeah, because you know, these were originally prokaryotes that we were absorbed into the eukaryotic cells, and then so these are called plastids. Shoot. Yeah, plastid. Yeah, plastid. Sorry. And the genes, so genes produce enzymes and proteins. Well, this makes sense. The genes code for proteins, and so DNA. I think it's. Hold on. Let's see. Genes code for DNA. DNA codes. No. There's genes on protein. Okay, so DNA and such genes code for proteins. Proteins do everything. All right. Let's see. So when sperm donates the DNA to the egg, and so the organelles come from the egg, which makes sense if you think about it. And then the mitochondria in zygote. So zygote is the after the egg and the sperm, you know, come together. And then it's an accurate way to identify parents. Let's see. So mitochondrial DNA is inherited from a single lineage. Interesting. Okay. I guess if you really wanted to see, this is what it looks like. 1,500 protein, a lot of proteins, mutations. Let's see. Okay, so we don't actually need to write. I didn't write any of this. We don't need it. Mitochondrial replacement therapy. Egg is removed. Blah blah blah. Okay. No, we don't need to read the rest of that. So now we're on mitosis. Wait. I think I meant to write meiosis. Yeah, not mitosis. It's meiosis. Alright. Meiosis and sexual reproduction. I wonder what this is supposed to be. What the freak? Oh. These were our last notes, by the way. This was the last thing we ever wrote. For notes. Alright, let's see here. This is not important. We're about to talk about all of this. Variations on a theme. Okay, so I wrote a little joke here. I said it's like Rhapsody on a theme of Paganini. You know, there's also variations of a theme of Corelli. There's Brahms also did Paganini. You know, it's a common musical thing. And then so living organisms are distinguished by their ability to reproduce of their own kind. So we'll talk more about evolution later. It comes up in there too. But for now, we're just going to move on. So heredity is the transmission of traits from one generation to the next. Variation is the difference in appearance that offspring show. So, you know, you might have the same genotype, but you might express a different phenotype. And then you have genetics. It's the scientific study of heredity and variation, so the combination of those two. And now we're going to talk about genes. So genes are segments of DNA that code for the basic units of heredity. And they're passed via the gametes, which are the reproductive cells. So, for example, in humans, that'll be the sperm, father, egg, mother. And then each gene has a specific location on the chromosome. So that's what I meant to say. It's called a locus. And then, so there's different types of reproduction. I'm gonna eat, so if you would like to read it, you can. But this is basically what's going on. This song is not period appropriate. It is May. John had a better effect. James, while John had a better effect on the teacher. Look what I just found. It's on Instagram. My friend sent me this. This is a grammatically correct sentence. Isn't that interesting? English is funny. Variations on a theme. English is very strange. Okay. Did you read through these? I hope you did. It makes sense. Asexual, it's not a sexual relationship. It's by yourself. Sexual, 2 parents. Little variation, so you can leave this like bacteria. Lots of variation, humans. They're faster and energy efficient because it's only 1. Slower, energy consumptive, but battery efficient. We're talking about bacteria. Yeah. All right. So meiosis is cell division, where the number of chromosomes is half to produce sex cells. So first, what it does, right, is it creates these copies, but then it creates, each of the end cells. There's 4 identical cells, or not identical. Sorry, there's 4 end cells, and they each are haploids. As a little review, somatic cells, so they're body cells, right? They're diploid, and they have 46 chromosomes. And then so there's a homologous chromosomes in each of them, from each parent. And then autosomes, so there's autosome chromosomes. There's 22 pairs of them. They don't determine sex, they determine all the other features of us. This isn't how biology works. Please read a book. I already studied biology. I'm just reviewing my notes. Thank you very much. Then there's the sex chromosomes. Okay, you want to see my notebook? I've been reading this throughout the year. I just figured I don't want to read through it again for the biology exam. Give a little bit back. Alright, anyways. So then sex chromosomes, X and Y, XX, XY, gametes. What the freak am I listening to? What the? Okay, we're gonna move on. Okie dokie, this is nice. I don't know if Nintendo will sue me, probably not. Eggs, sperm, XX or Y, Okay, so diploids, total number of chromosomes, consists 2 sets. We've already talked about this. Haploids, I'm not going to stay on here that long. Egg cells, you get half of it from your mom, half of it from your dad. Homologous chromosomes, they're the same length. They, the same genes are in the same location. And we have, this will be important when we talk about Mendelian genetic variations. So, yeah. You want to look at it here. Where are we? Not there yet. Oh, okay, we're still on this page because we haven't gotten to the actual prophase 1, prophase 2, that sort of thing. Does this make sense? So you have homologous chromosomes. Right, they're the same genes for different alleles. Non-homologous, they're different. And then sister chromatids, they're the same gene and genes and alleles that help to get the centromere. That's what this example I was talking about earlier. One example. See. Now note Y is much shorter than X. I don't know if you actually need to know that. Autosomal chromosomes, all chromosomes, blah blah blah. Humans have 22 pairs. So this is what we call a karyotype. That's all of the chromosomes. And so you'll notice Y is significantly shorter than X. Two divisions. Meiosis 1. And then we'll switch them into their haploids, or into their diploids, or haploids, yeah. And then meiosis 2, which then, again, switch them into their haploids. But what they each do is different. So we'll start with prophase 1. So, and then this is where we talk about genetic diversity. So there's crossing over, which is basically when, like, the ends of the chromatids, they cross over and they exchange genetic material between homologous chromosomes. Protein lattice, I guess you need to know this. Protein lattice appears between chromosomes, ensure they're aligned. And then now the chromatids are no longer identical, right? Because we've swapped. And so this increases genetic diversity, obviously. But this is a visual, see? Recombinant, basically it's when you put different parts of DNA together. Pardon me. you said please read a book. I'm not gonna take that personally. Metaphase, we're still in 1, by the way, so as you remember, there's 2 meioses. So metaphase 1 is where the homologous chromosomes are on the metaphase plate. And then independent assortment, so basically what this is saying is that there's no certain way that the homologous chromosomes have to line up that way. And so they can separate differently. And then, so you'll see, like this, see? There's different combinations by the way these can line up. And you see, even just for 3 pairs, there are 8 different combinations that could occur. So I was reading that fact. It is really true. It's really stark, the difference. Exponents. A lot of combinations. Now, anaphase 1 is where the homologous chromosomes are pulled apart. So you see, including the crossing over stuff that they did. Crossing over occurs. It's supposed to occur in prophase, but the sister chromatids remain attached. So now you'll see. They're still diploids because there's not 2 of each chromosome. But yeah, interkinesis, rest period, I guess that's what it's called. And so it doesn't go through telophase, I guess. Oh no, it does go through telophase. I'm sorry, I don't know what I'm talking about. And then so at the beginning of meiosis 2, the cells are now haploid, right? Because they still have their sister chromatids. But then now they're going to... So the first step is splitting the homologous chromosomes, separating them, and then the third step... No, so the first step is splitting homologous chromosomes. The second step is now we're splitting the sister chromatids. Chromatid. Okay, we're still on this page. Alright. So as you can see, same thing again, very similar to mitosis. It's just the functions are different. This is just a summary. Because, so if it's an identical copy, right, then a mutation has to occur in order for some change to happen. But since we have the crossing over and independent assortment, it's a lot easier for those for the diversity. Better chance to survive. Main goal of sexual reproduction is to create a larger diverse of organisms. Okay, now we're getting close to Mendelian. I think we're about to go to sexual reproduction. Okay, if you want to read this. We'll see how much we can get done in about 30 minutes. Because I have an event I have to get to. This is good. I'm gonna change the music though. I like this one. Okay. Read this? Yeah. Okay. Sexual reproduction of a mother and a father, 2 individuals. So, see, you have 2 gametes, and they form a zygote. If you want to know what it looks like, this is what it looks like. I'll let you infer what parts of the body these are. Alright, cycle of life. Fertilization. Single sperm, an egg. See? So if you think about it, you competed against, I don't want to say, if your sperm won out of like the hundreds of thousands that were, competing for the zygote. So congratulations, you're lucky that way. I already told you what a karyotype was. Yeah. Please read a book. Maybe I'm gonna be a little too personal. Sorry. Alright, non-disjunction, so failure of chromosomes to separate, that results in a loss or gain. And then, so we'll talk about those here. Euploidy, average number in species, and aneuploidy, changing the chromosome number. What did we need to know these for? Oh, because now we're going to talk about monosomy and trisomy. I guarantee it. Yeah, okay. So monosomy is where you have, let's see, 2n minus 1 occurs when an organism has one of the two pairs of homologous chromosomes. So you can see here, right? So we have one less. And then trisomy is when an individual has 3 instead of 2. So only 3 trisomies are viable beyond birth. Interesting. So then you see here, right, you have extra. This one you have not enough, this one you have too much. And images containing not so individual. Okay, so now we're talking about specific, we don't need to know these. I guess I'll show them for the sake of education. Alright, so if you have a trisomy 13, and then so these are the only trisomies you'll survive. Patau syndrome, major eye ring defects, rarely live to live long. You have 18, and then so Edwards syndrome, almost all organs are affected. You have a small heart, rarely live more than a few months. 21, most common type, Down syndrome. I'm sure you know what Down syndrome is. Okay, and then so this is it. So females, let's see. So you either have an so this is like what it looks like if it's wrong. So this is not disjunct. So if you have 2 X's, so then if you have an XXX, it's just female. If you have an XXY, it's a male with a syndrome. XO, you have Turner syndrome, and then just the Y sperm itself is non viable. You need at least one. So we have Turner syndrome here. If you know, that's when you only have one X. And then so you develop as a girl. What do you call it? You have physical irregularities. Shorter life, you're infertile. So, and if you have XX and a Y, so you have it's Klinefelter. So you develop as a male. Apparently, in a lot of cases, go undiagnosed. Incomplete puberty, less muscle, less facial hair, and less breast tissue. Osteoporosis. Ouch. And XXX, triple X. You develop as a female. Interesting. This doesn't look that bad, like if we're relatively comparing. Now an XYY, so you develop as a male, physical irregularities, you're taller than average, slight female. Okay, so you don't need to know these. This was just the point of learning. Can you? Let's see. Okay, and then so these are primary sex characteristics. Oh, you also learn these in psych, primary and secondary. SRY gene is individual XY is deleted, it will develop as a female. If the SRY gene translocates to a person who's XX, they will develop as a male. The joke is there is no next slide. Okay, so you see, right? So this is an SRY gene. So it stimulates the growth of the male reproductive system. So hormone testosterone changes physical features to male. So then you'll notice if you put on a female, the SRY gene, then you become you develop as a male. If the SRYG is mutated or missing, then you'll be an XY female. All right, I think yes, this is the next thing we're talking about, which is Mendelian. All right, you already talked what a gene is. We know what an allele is. This is the genotype. Let me set an alarm so I don't forget. Set a timer for 2:25. Okie dokie. Normally I'm just eating some rice. Okay. Now, genotype. So it refers to the alleles an individual receives a fertilization. So, for example, AA, you've probably seen these before. When we talk about, I'll point it out again when we talk about Punnett squares. Now, the phenotype is the physical appearance that's based on the genotype. Now, the important thing to know is that you can have the same genotype, like I mentioned, but you could have a different phenotype. All right. Ooh, yeah. Stretch it out. I heard myself downstairs. I think that's pretty funny. The song's too calming. We need something more. Yeah. Bum, 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 bum. Yum, bum, 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 bum. All right. Homo, hetero, know the difference. Homo means same, hetero means different. So homozygous means it'll be the dominant or recessive. So you have homozygous dominant, where both of the alleles are dominant. You have homozygous recessive, which means both of them are recessive. Heterozygous means they're different. Usually when you write them, they'll just appear as dominant recessive, but I mean, if we're talking about like alignment locus, then that's the way it can be. All right, so the dominant allele, so let's see, produces fully functioning protein, and the recessive produces a protein with little to no function. So that's, I guess, a scientific way to think about it, but basically the dominant overpowers the recessive, if it makes sense. Law of dominance, only one form of the trait will appear in the next generation. So as you can see here, homozygous dominant is going to be red cherries. Homozygous recessive is going to be yellow cherries. But heterozygous, because there's one dominant allele, then it's going to express the dominant phenotype. Therefore, it's just going to be red. But then there's other ones. Heterozygotes, just allele uneven. That's allele funny. Get it? Anyways, so now we're going to talk about Gregor Mendel. He was this monk. 19th blending, some evidence to not support, or some evidence to support, however, some evidence disproves hypothesis. Two pink flowers producing red and white flowers. So blending concept of inheritance. So I guess this is what it was before. So you have like a smooth and a wrinkly, but then Mendelian's results was you get a smooth wrinkly and then you get smooth. All right, and then so this is a pedigree. This is what this is called. Usually it'll just be circles, squares, shaded or not shaded. I guess this is a variation on that. Yeah, well, Gregor Mendel, this is the guy, did all the stuff. Vienna, great place. I've never been, but I love their orchestra. A few scientists apply math to biology, just... Okay. Chose pea plants because the short generation, it makes sense. Humans have long generations because we have long lifespans. And it takes a long time to reproduce. Oh, so I guess this is how. Oh, and then this was on my test. I don't know if you need to know this. So the stamen is the male part, and so you collect the pollen, sperm, and then you give it to the female part, the carpel, and then, yeah. Mendel made sure plants were all true breeding. Mendel chose plants only varying in one trait. This is how you control the variables. Okay, now these are just some vocab. P1, parent generation. F1, offspring of parent. F2, yada, 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 goes on from there. And so these were his results specifically. Because note, a tall and a short will create all heterozygous, right? It'll create all heterozygous stuff. And then so therefore the next generation, if you cross a heterozygous and a heterozygous and have 4 offspring, what you can expect is you're going to have 3 of them. You're going to have one that's homozygous dominant. You're going to have 2 that are heterozygous. You're going to have 2 that are heterozygous, and then you're going to have one that's recessive. But note that according to the law of dominance, the heterozygous and the homozygous dominant will express the tall gene, which is why you see a 3-to-1 ratio. This is important. This was on one of the FRQs that I took for my final. Proposed particulate theory, my time will come. What does that mean? Okay, now we're talking about Punnett squares. See? So these are the genotypes. I forgot what I said I would mention before. This is dominant, this is recessive. Yeah. I'm not going to teach you how to do a Punnett square. We're like a day away from the test. So a monohybrid is just a cross of a single trait. So it's like, those will be your 2x2s. Punnett squares, so letter outside. Oh, you can see it's covering sperm, sperm, egg, egg. Okay, and then so the answers are, if you wanted to try, you can try. I'm not going to show you to though. There's a law of segregation, which basically is saying each organism has 2 factors for each trait, and the factors separate during meiosis. That's why there are haploids. And then each gamete only contains 1 of the factors. Punnett square, so it was Mendel. Interesting. So, yeah. What did Gregor Mendel say when he founded genetics? Whoopee! Yeah. Alright. Mendel began to wonder what would happen if he studied plants that differed in 2 traits. Blah, blah, blah. These are just questions. Locus, we already explained. Location of a gene, the homologous chromosomes. Law, and then they can have the same location, but they'll be homologous chromosomes if they have different alleles, and they'll be sister chromatids if they have the same allele. Law of independent assortment. Each factor segregates independently, you see. And then, so a dihybrid cross is a cross between 2 different traits. So you have the A and the B trait here. And then for that, you need to create a 4x4. And so the way you do it, see, you figure out all the different gametes. So what you would put in the squares, you would put in A, capital B, A, lowercase b, A, lowercase a, capital B, lowercase a, lowercase b. And you would put those in these slots, and then you would cross. So like that. See, and then these are all the possible gametes. Make sure you note this wasn't FRQ. Had a ton of FRQ. I think we've had like way too many for me to count. We've had a lot of them. Okay. So I'm just what you would write down here. You have an capital A, capital B, capital A, lowercase b, lowercase a, capital B, lowercase a, lowercase b, and you have the same here. And you would just write down all of the genotypes. And then you would figure out the phenotypes and figure out the ratios. Now we're talking about probability. That's fine. Dihybrid, just no probability. It's like, for example, if you say, what's the probability of getting this or that, you would add these 2 together. But then you're saying, what's the probability of getting this and that? And you would multiply. We're not going to talk about that. Okay, there is a little trick. So, like, let's say you get into these larger ones. What you can do is you can just find the probability of each individual occurring. So you just take each trait and then you find its individual probability to then do that. Okay, now we're going to talk about human genetics disorder. We're almost on recombinant DNA, which means we're getting really close to the end. Unfortunately, I won't make it this stream. I didn't lock in hard enough. But that's okay. Let's see, so autosomal disorder genes found on chromosomes 1 to 22. So it's just a disorder with any of these. Autosomal recessive, so you need to have both of the recessive genes to have the disorder. And then so you can be a carrier because it's just heterozygous. So they do not have the disorder. Because the dominant will take, the law of dominance, it'll express itself. Apparently it's rare. CFTR genes. Okay, so this is an example. Cystic fibrosis. You don't need to know these. We're just, little knowledge for your noggin. Human genetic disorders, autosomal. So now you have autosomal dominant. So anyone with the gene will have it. So it doesn't skip generations. Huntington's disease. Genetic recombination. Chromosomal theory of inheritance. Chromosomes are inherited from parents. Genes for traits are located on chromosomes. It's what we've been learning. This is unrelated to it. These are 2 different photos. Studied fruit flies. Ratios didn't make sense. So, genetic recombination. When I blew off his homework, figured out crossing over. Really? Okay, so recombinant DNA is where genes cross over. So you can see they're like recombined. Moving on, linked genes. So they're genes located on the same chromosome. So for example, you see a set of genes. They're likely to be inherited together. Parents just got home. Okay. genetic recombination. So, like this is an example: red hair and freckles. Unlinked genes, so the genes that are different. Do we learn about it in this one? We do. The locations of them. So I'll show you what I mean. Crossing over frequencies. So this is where you create the crossing over map. Or we could keep talking about this. Genetic recombination, production of offspring with combination of traits different. This is scary music. We're moving on. Okay, we'll do this one. Offspring looks like parents. If different from parents, they're recombinants, so you can see. And then, so this is how you figure it out: the number of recombinants minus the total of offspring, and then it should add up. Now, when we're talking about this, what this is 25. I got 5 minutes. Let's see if we can finish the chapter in 5 minutes. I'm pretty sure we can. Chromosome 50 map units long. So here's an example here. So you see crossover frequency. So we see it's the largest, right? So here, let's actually open this up on a whiteboard so I can demonstrate. Not that. I have the app. Yes, open it, please. Alright, so it's going to be with a mouse, so it's going to be a little bad. But let's say you have this, right? So if we use this as an example, and then so what we can see from this photo is that the largest difference is between B and C. So what we're going to do is we're going to place B here, and we're going to place C here. And then now we see that A and C has a difference of 6%, and then A and B has a difference of 12. So technically this would be split into thirds, because then you see that this is the 6%, and that here is going to be the 12%. So basically this is what's happening. That's how you do it. It gets a little more complicated when you have larger ones like this, but it's easy. So you put B and C on the outside, and then you put B and D. So that means the difference between C and D should be 5%, and then you have, yeah, you can figure out the orders. It's simple once you think about it. And then to figure this out, basically you would just do 244 divided the total. Non-Mendelian genetics. So multiple allele traits. So basically it's saying that traits are controlled by multiple different alleles. So like for example, you can see here, like you have the dominant, but then multiple of these recessives, there's multiple different alleles for them. And it's usually not that simple. It's usually not Mendelian simple. That's what I meant to say. Let's see. C, CCD, CCL, CH, C, for example. And then so if you wanted to find a cross between a gray rabbit, so that would be I believe CL, because it says you're albino, CL and C, and then a black rabbit, which is C and C. So you would have at least one albino. You can do that on your own. And then so there's these two different kinds. These are non-Mendelian genetics. Last one, incomplete dominance. It's basically where both are partially expressed. So there's an intermediate phenotype. For example, if you cross red and white flowers, you'll get a pink flower, or in this case, you get a green monster thingy. And then codominance is where both phenotypes are expressed. So you can see there's blue and yellow expressed. Not like an intermediate between the both. So these are examples. Yeah, see what I just said? Pink flower. And then here's a codominase example. Multiple alleles. Okay, now this is for blood. This is something you probably should know. We're getting pretty close. On the last page. Let's see. Neither antigen allele. If you have neither, let's see. So antigen is either sugars or proteins that are attached to various compounds in the red blood cell membrane. They identify self versus non-self. Okay, we're about to talk about those, so let's get there. So you know there's A, AO, BB, BO, AB, or OO. So I'm assuming O is recessive here. Let's see, antigen, either sugars or proteins. Okay, so these are antigens on the red blood cells, and then so I guess they help identify like the type of blood. And then antibody protein producer immune sec. I've already written this down. We are running out of time. I have 2 minutes. Okay, so you can see this is positive blood. So they have the positive antigen, right? And then that's why these are compatible with positive, but the negatives are not compatible with positives. But negatives, so positives and negatives can't be together because of the antigen. But you can't donate a positive to a negative, but you can donate a negative to the positive. That's fine because of the C. Notice the negatives have that antibody, and then anti-B, anti-A. So they don't work together. But then these have neither of the antibodies in their plasma, but they do have both of the antigens. O is the universal donor because it has no antigens to react with. If you look here, there are no antigens. Nothing's on the outside, by the way, if you're wondering. And then AB is universal receiver because they have no antibodies to react with foreign blood. So it can take in any of them. I have 1 minute. RH factor, more antigens. Let's see. So positive for French presidents, homozygous or heterozygous, negative, no antigens. A woman is heterozygous. Okay, so these are just more practice. This is the percentage. There's a lot of O+. Sex-linked genetics. These are for the last pair of the chromosomes because 22 of them are autosomal. Sex-linked gene, any gene that is located on the sex chromosome. In humans, most sex-linked genes are found on the X chromosome. Blah, 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 blah. Let's see, sex-linked recessive. So recessive, it's going to be, recessive. I don't know how to describe that more. Males will only need one copy, females will need two, because if you think of it, males only have one X chromosome, and so if they have it on that chromosome, then they will have it, but females have two X's, so they need it on both. Carrier, we've already explained it. This is what a pedigree is. It's just with a more complicated shape, that being XY. Interesting. Affected males can only receive disorder from mother. Let's think about it. Why? I don't have time to think about it. Okay. Bad vision. I can't imagine. I feel bad. Sorry. 74. Sex-linked recessive. Okay, we don't need to do that practice. Dominant. It's where, again, males only need one, but I guess they can receive it from the father. And then the females, they only need one too, because it's dominant. Y-linked disorders. Oh, look at that hair. So it's always passed from father to son. It makes sense because it's the Y gene. Males only have this disease. Makes sense. Females don't get it because they don't have a Y chromosome. Okay, probability. Don't know why we're talking about that. Polygenic inheritance. Oh my goodness. I thought we were close. Or we are close. Several genes contribute to a single phenotype. Yeah, that makes sense. Think about it. If multiple genes code for a single trait, several genes will contribute to a single phenotype. Polygenic inheritance. A practice question. This is fine. Okay, so this is what you need in order to do a 3-trait Punnett square. It's not that complicated, really, to set up. So, skin color. It appears we're covering it up. Many genes contribute to skin color. Okay. Incomplete penetrance. An individual may have the genotype but won't produce the phenotype. Interesting. Variable expressivity. The range of signs and symptoms that can occur. Okay, so this is like either you express it or you don't. This is you can express it. So it's like it's either a 0 or a 1 if we're talking about incomplete, but then it can be like a 0.0 to a 1.0 if it's variable, like a brightness. This is just an example. This is also an example. What is this? Occurs when a single mutant gene affects 2 or more distinct and seemingly unrelated traits. Did you know that the dwarf gene is dominant? I did not. Epistasis, interaction of 2 or more gene pairs at different loci influence the same trait, but one allele has an overriding effect. Gene A controls gene B. The gene B influences the effect of the gene A. The gene B is said to be epistatic to gene A. Okay, so I guess this should be switched, or is controlled. So hair color. Interesting. Golden is recessive. Pigment control turned off. E turned off. E. Requirement. Okay. Interesting. Chromosomal mutations. Okay, so this, I think we talk about this later, but we'll just talk about it real quick. Deletion, you delete a segment of the chromosome. Duplication is where you duplicate a segment of the chromosome. Inversion is where you switch the order or versus a segment. Translocation is where you move from one chromosome to a non-homologous chromosome. Don't get those confused. Okay, pedigrees, real quick, not the food. Family tree, so you can see female, male, shaded in is usually the trait. I guess half if it's the carrier. Yeah. Question marks can be only correct answers. What? Okay, marriage line, line of descent, sibling line. Okay, so you just need to know what that stuff means. Autosomal recessive. So then, for example, if it's autosomal recessive, that means you need both of the letters. And then so we can assume these two parents are carriers, and therefore this one will have it got unlucky, I guess, or lucky, depending on the trait. Autosomal dominant, it doesn't skip generations, so you need at least one. And then these two could still be heterozygous. And then, so that's why he's ineffective. As you see, parents must be heterozygous, parents must be heterozygous. So you have X-linked, and then so it says sex linkage cannot be confirmed, X-linked dominant, but you can confirm Y-linked because they're always passed from father to son, and then it will only affect sons. Okay, we are almost there. Environmental factors, plasticity, phenotype. The environment, your body, you can change depending on your environment. Complexity increases as you learn more about the subject. There's so many. Okay, so this was just our teacher reminding me to stay strong. Okay, that is it for now. We don't have that much left, actually. We did get through a lot. So maybe if I have time, I'll resume because the test is tomorrow. But if not, not a huge deal. Alright, thanks for tuning in. Good luck on your AP test tomorrow.